is a professor and a coordinator at Dr. B.R. Ambedkar Center for Biomedical Research, University of Delhi. Her area of interest is in molecular modeling, drug design, and bioinformatics. Professor Chopra did her PhD in chemistry from Department of Chemistry, University of Delhi. She is also a life member of several scientific bodies in India. And she also conducted a drug discovery hackathon for open source drug discovery for COVID-19. The second chairperson for this session is Dr. Manoj Kashyap, who is coming from Faculty of Health and Allied Sciences from Amity University, Manesar. His area of interest is in genomics and proteomics of gastrointestinal malignancies. Professor Kashyap did his PhD from the Johns Hopkins University in the US and further pursued his postdoc from Moore's Cancer Center at University of California. Professor Kashyap is member of Royal Society of Chemistry as well as American Chemical Society, and he also serves as a reviewer and editor on several journals. So now I request Professor Chopra and Dr. Kashyap to initiate the session. Thank you.
Very good afternoon to everyone. Though we are delayed, but 2 of the talks have been cancelled, so we have enough time for this session. So first of all, I would like to introduce the first speaker, Dr. Satyanarayanan. Dr. Satyanarayanan is Principal Medical Superintendent and Director R&D at Munial Institute of Ayurvedic Medical Sciences, Manipal, Karnataka. His area of interest includes medicinal plants and herbo-mineral formulations, quality control of herbal and herbo-mineral formulations, biological standardization of Ayurvedic products, etc. He is a BAMD from SDM College of Ayurveda, Udupi, India, and MD in Ayurveda in Bhavishya Kalpana from Gujarat Ayurveda in Jamnagar. He has published more than 20 research papers in national and international journals, and of course some best paper awards in Colombo, Sri Lanka on Gary International Conference. He has been conferred with Edward Kennedy Memorial Award for Best Presenter in a senior research category. He has experience of 21 years in the subject Rashtrastra and Bhavishya Kalpana. He has worked as R&D executive at Munial Ayurveda Research Center. Today, his title of the talk relevant to this conference is Application of Ayurvedic Herbals and Formulations in Cancer Chemoprevention and Radioprotection. With these few words, I welcome Dr. Satyanarayanan and to give the talk.
Thank you. Shankham Chakram Jalokam Dadhamrita Ghatam Charadurбhishchaturбhiः Sukshma Swachhati Hridyam Shukaparivilasan Maulimam Bhojanetram Kalam Bhodo Jwalangam Katitata Vilasat Charupitam Varadhyam Vande Dhanvantarim Nikhilagadawana Praudhadavagnilelam. So let us pray Lord Dhanvantari to destroy all our enormities and bestow us with the best of health. Good afternoon. At the outset, let me thank the organizers for giving me an opportunity to stand before you and share my views on the topic that is displayed here. Respected chairpersons, august delegates who have joined me physically and virtually. The topic that I have selected today is on Ayurvedic medicinal plants and formulations in cancer chemoprevention and also chemoprotection and radioprotection. So let me begin with some customary slides. We know that cancer is the second largest cause of death worldwide. Today, even after so many advancements in the field of oncology, maybe in diagnostics or in therapeutics, it has still remained a challenge in healthcare. So based on my clinical experience of last 22 years, I'm very much confident to say that being a member of Ayurveda faculty, Ayurveda has a lot to offer here. I'm not going for the details of those because my focus of today's presentation is on only chemo prevention, protection, and radio protection. We have seen that there are advancements in cancer detection methodologies. Maybe newer drugs have been invented for cancer chemotherapy. Nanotechnology has been adopted very well. And new techniques of radiation, where focused linear radiation is being followed and immunotherapy is becoming popular. Even then, we are seeing that excruciating patients of cancer are running towards alternate paradigms. So in my experience of last 2 decades, we have a data of over 1,000 patients who have visited us maybe in virgin status or in terminal status. And we had a lot to experience by interacting with these people. We know that there is a conventional mode of treatment in cancer, maybe surgery, or maybe chemotherapy, maybe radiation, or a combination of this one after the other, or immunotherapy as the next line of treatment. Many of the situations, not because of economic constraints, but because of adverse effects, which is so unbearable to the cancer patients that they were stopping treatment in between. They were running away from the hospital and they are trying to find out some other solution for the problem. We, being the people of Ayurveda, have definitely an important role to play here, because role of Ayurveda is not simply prophylactic. However, when we talk about Ayurveda, we speak about diet, lifestyle management, all these. Definitely they have an important role even in cancer prevention. You know very well that by proper diet and lifestyle management, you can influence epigenetic markers. So you can make those particular loci of the genetic picture not to get exposed, or the cancer not to manifest, because we cannot change the gene, but we can change the genetic expression by the epigenetic influence, which is definitely possible with Ayurvedic lifestyle and also food. So prophylactic role in cancer is definitely there. There is no doubt about it. We have seen that in a few cases, we have a curative approach also. At the same time, supportive and palliative role of Ayurveda can never be neglected. So based on this, we had developed a comprehensive approach, a multidimensional approach entitled Mahaushadha Kalpa in our institute, for which we have received a US patent also, which is not simply a research drug. It has got a drug, it has got formulations, it has got diet and lifestyle management, it has pyramid therapy, it has yoga, it has pranayama, it has supportive procedures like panchakarma, etc. We have always seen that cancer can never be managed with a single approach. It should have a comprehensive multi-dimensional approach to get best possible yields. Today, I'm not going for those details of our approach to treat the cancer, because this seminar is mainly focused on the diet or dietary supplements or those simple herbs or formulations which can be used over the counter to support cancer. I'm not talking about those potent formulations or other methodologies, because it is out of focus of today's lecture. So even during radiotherapy of cancer, it is very important for us to protect normal tissues. However, now the instrumentation has improved, the methodology is improved. Despite that, we are observing that there are deleterious effects of radiotherapy, especially in oropharyngeal cancer. You must be knowing that in oropharyngeal cancer, almost invariably radiotherapy is used, maybe post-operative or as a first line of treatment or concomitant with chemotherapy. In such situations, there will be sometimes a permanently damage, like a salivary glands will be affected. Dryness will be there throughout the life of an individual, cannot produce saliva. There will be dryness of the mouth. Oral mucositis is very common. Dysphagia sometimes may be permanent in such situations. In other areas, suppose there is C of esophagus, if it is somewhere midway, then it becomes extremely difficult because heart is very much close. So radiation becomes very difficult in such situations. Or post radiotherapy, there may be strictures in esophagus, which may have to be dilated repeatedly also. There are many other issues related to even radiotherapy also, which many times we feel as very simple. There is an interesting statement here in the last line. You can see that recent advances in the area of radio protection have shown that it is possible to modulate the radio response in normal and tumor tissues by making use of natural plant products. You can use certain plant products which make your tumor tissues to be more responsive to the radiotherapy. The tumor regression will be more pronounced by radiotherapy when you use such supplements concomitant to radiotherapy. And also some of those studies which have been published have shown that when you use such herbs, normal tissues have been protected better, and also those tumor tissues became more susceptible to radiation. That is another very interesting application of natural products along with the regular radiation. What can be the role of medicinal plants in cancer treatment? The plants can have their role in fighting cancer by various mechanisms. It may be simply nourishing the body with the minerals, vitamins, or some natural enzymes. You know that we have papain, bromelain, like natural enzymes that are present in the plant, and also micronutrients. And most importantly, immunomodulating effect, the increasing immune system of the body, or inducing antioxidant action and protecting the body from oxidative stress. The majority of the plants that we use in Ayurveda are having free radical scavenging properties. They enhance the detoxification function of the body. They alkalize the body fluids. Studies have shown that by changing the pH of body fluid, you can fight cancer to some extent. They inhibit some cancer activating enzymes. They promote production of protective enzymes. They may stimulate DNA repair mechanisms, and also they modulate the activity of specific hormones and enzymes to inhibit the growth of cancer. These are a few of the mechanisms. There can be many more mechanisms by which a plant or a plant constituent or a polyherbal plant product can act. There are many studies which have reported that phytoconstituents, maybe the flavonoids like quercetin or catechins, which we have in the drugs like Acacia catechu, or resveratrol, which we have in our plant products like Draksha Rishta, Draksha Sava, etc. Green tea polyphenols, even though we do not use them in Ayurveda. Grapeseed proanthocyanidins, silimarin, curcumin, about which today morning we had a very important session. And I think extensive study has been carried out on anti-cancer potential of curcumin. Even though we are not using isolated curcumin, we may be using turmeric as a whole, but we use a specific medium in which that curcumin is soluble, and it will show the action. If you just make a decoction of turmeric and use it, it may not be efficacious. But if you boil it in milk, it has got a typical binding property with the protein and fat there. Then only it will be bioavailable and it will show its action. So that beauty is known to Ayurveda, and accordingly a specific mode of administration is described. These are all the plants or plant products which have anti-inflammatory and immunomodulatory properties. So there are many chemopreventive agents which are phytochemicals or secondary metabolites where plant may not need them as a nutrition, but they are produced as a disease preventive property for us. So many a times this is a tricky situation where many of the pharmaceutical companies fail in the quality control of their products. we know that Ayurvedic industries mainly depend upon the forest or natural resources for the plants. In forest, plants grow not in a scientific manner, in a natural manner, where a plant has got a stimulant nearby. It has got a typical sympathetic ecosystem. If you try to cultivate the same plant in a field, say 10 by 10 feet, and feed all manure to it, plant may grow very well, but it may not produce certain secondary metabolites which are required for therapeutic action. So some studies have shown that the plant like Vasa Ardhoda Vasica, when it was collected from 10 different places, vesicin concentration varied to a great extent. In one of the sample, it was not at all present. So there should be some natural phenomenon or surrounding environment for the plant to produce these secondary metabolites. However, nowadays artificial stress is being induced to the plant in tissue culture method, where the plant is made to produce such phytoconstituents. That's a different story. So these are the things which make Ayurveda a bit tricky, and standardization very difficult, but you should have an insight to the idea that our classics have mentioned to maintain a quality in these medicinal plants. So during the initiation phase, natural chemopreventive agents, probably they inhibit the absorption of a carcinogen into the organism through antioxidant activity. They may prevent inflammatory induced damage or enhance neutralization, and they remove carcinogens through metabolic enzymes. There are several medicinal plants, just to quote a few: Curcuma longa or turmeric, Osmum sanctum or our Tulasi, holy basil, or Tinospora cordifolia, about which we heard today morning, Guduchi, in Hindi known as Giloy. They all have been evaluated extensively for their anti-tumor, antioxidant, and chemopreventive effects. There are many classical formulations of Ayurveda like Amrita Prasham, Ashwagandha Rasayana, Brahma Rasayana, Chavana Prasha, Narasimha Rasayana, Rasayana Avaleha, or well-known Triphala, which belong to a group of drugs known as Rasayana. We were expecting a talk on Rasayana in this session. I think the speaker is not there today, who would have thrown more light on this concept of Rasayana and different modes of action that Rasayana plants have. So these are the drugs that impart rejuvenating effects to the cells and tissues, and they are all reported to have radioprotective effects. So let me just give you a few examples today, just hand-picked examples, radioprotective Ayurvedic herbs. To start with, Mandokaparni or Centella asiatica, it's called as Gotukola in English. Many people use this as a Brahmi. This is not Brahmi. Brahmi is Bekapa Moniri. This is a Mandokaparni. Sometimes it is used as a substitute of Brahmi. The therapeutic properties slightly vary. It's not exactly the same. This is used as a, for in cuisines also. We prepare chutney and use this in southern part of India. Aqueous extract of Centella asiatica reduce the adverse effect of low dose irradiation in Sprague-Dawley rats by inhibiting radiation-induced body weight loss and also conditioned taste aversion, according to Shobivi et al. And Sharma J et al. Have reported that it also has found to protect against the radiation-induced weight loss in mice exposed to 8 gyri of gamma radiation. Tulasi, holy basil, is one of the wonder drug which we use in multiple conditions in Ayurveda, maybe as a single drug or in combination or in different forms, various forms. It has got a well-proven radioprotective property. Probably it was first reported by Jagetia et al. Against the radiation-induced mortality. So thereafter, studies by Umadevi and her co-workers established its radioprotective efficacy by evaluating mouse survival, then spleen colony assay, and chromosome aberrations in mouse bone marrow cells. Apart from these, Tulasi has been reported to protect against radiation-induced lipid peroxidation and reduction in glutathione concentration. Baliga MS and others in their review article have mentioned that Tulasi and its flavonoids selectively protect the normal tissues against lethal effects of radiation. Tulasi also protects against radiation-induced damage to the salivary glands, which I was speaking about, which is a very common adverse effect of radiation in oropharyngeal carcinoma. Flavonoids of Tulasi protects against radiation-induced gastrointestinal damage in several GI tumors, even in colon carcinoma. Radiation is used sometimes. Tulasi extracts its flavonoids and one of the important phytoconstituents, eugenol, protect against radiation-induced clastrogenesis. Tulasi also enhances bone marrow radioprotection with WR2721. So WR2721 is nowadays being used for radioprotection. The Tulasi was compared with that separately and in combination. They have found that in combination they have got excellent radioprotective effect. Tinospora cordifolia, which became immensely popular in general public during COVID pandemics. However, for us Ayurvedicians, this is a well-known drug. We call this Amrita. The name Amrita itself is because of its therapeutic potential. Tinospora cordifolia, ideally its stem has to be used, fresh stem has to be used for best therapeutic results. However, people use its leaves also, which may not be having expected therapeutic results. However, it has got some specific therapeutic indication also. Oral administration of aqueous extract of guduchi has been reported to increase the survival of mice exposed to radiation. Hydroalcoholic extract of the same plant has been found to protect against radiation-induced micronuclei formation and also oxidative stress and decline in mouse survival, spleen colony forming units and hematological parameters. Our amalaki or amla, rich in gallotanic acid, vitamin C, has been extensively evaluated. You will get hundreds of articles on therapeutic potential of amalaki. The fruit pulp of amalaki or Emblica officinalis has been reported to increase the survival and inhibit radiation-induced weight loss in mice. Bhumi amalaki, Phyllanthus niruri is the plant that we use. Even Phyllanthus amarus is also used. Even this is also used in cuisines in our place. A well-known hepatoprotective drug. Most of Ayurvedic proprietary products that are used as hepatoprotective or in various types of liver disorders do contain this excellent antiviral drug, even anti-tumor drug also, Bhumi amalaki or Phyllanthus amarus. So this also has been reported to protect against radiation-induced decline in WBCs, decline in natural antioxidants like superoxide dismutase, catalase, glutathione S transferase, glutathione peroxide, and also glutathione reductase. Piper longum, commonly known as long pepper, one of excellent bioenhancers. If you see 100 of Ayurvedic formulations, 90% of them do contain trikatu. One of them is long pepper, mainly because of piperine, which is an excellent bioenhancer. If you add this with any other drug, its therapeutic potential is enhanced. And it is true even with the other chemical drugs also. You may be knowing that this has been tried with anti-tubercular drugs, other anti-cancer drugs, and it has shown bioenhancing effect also. So I'm not talking about that. So my concentration is on the effect of pipali as a radioprotective agent. Ethanolic extract of this was found to protect mice against radiation-induced decline in WBCs, bone marrow cell alpha esterase positive cells, and also GSH. Pipali extract also reduced the elevated levels of glutathione pyruvate transaminase, of course, ALP and lipid peroxidation in liver. It's a very good hepatoprotective already, and serum of irradiated animals. Or common ginger or shunthi. Administration of about 10 milligram per kg intraperitoneal. I'm of course much interested in oral administration. 250 milligram per kg orally. Hydroalcoholic extract of this shunthi consecutively for 5 days could protect mice against radiation sickness, gastrointestinal as well as bone marrow deaths. Ginger has been reported to increase glutathione, reduce lipid peroxidation in vivo, and scavenging of various free radicals in vivo and in vitro both. Jambu, in Hindi known as jamun, Syzygium cumini. We use its leaves and seeds, even the bark, for therapeutic purpose. for various different indications. Here I'm talking about the scales, that is the bark of this particular tree, Eugenia cumini or Syzygium cumini. It has several medicinal properties. The micronucleus study of radioprotective effect of dichloromethane and methanol 1:1 extract of jambu was studied. In human peripheral blood lymphocytes, it ascertained that it has got radioprotective potential. Even in vivo evaluation also established its protective potential. It was found to reduce radiation-induced sickness and gastrointestinal and bone marrow deaths. Bilva, a sacred holy plant for us, for Lord Shiva. We use its leaves, we use its fruits, we also use its roots. Actually, Bilva root has to be used, one among Dashamulas. Dashamula you might have heard. But procuring Bilva root is difficult. You will have to kill the plant. You have to uproot the plant, or you will have to take only a particular part of the root, which becomes very difficult. So nowadays, most of the companies, they substitute roots with stem or stem bark or the twig for therapeutic potential. Anyway, for radio protective activity, it is the leaves that are found to be more efficacious. In a study, it is shown that the hydroalcoholic extract of eagle marmelos or bilva protected against HPBLs, where radiation has caused the generation of micronuclei in a very small concentration of 5 microgram per ml. The radioprotective activity of 15 to 250 milligram per kg of eagle marmelos extract was further confirmed in vivo studies, where it's intraperitoneal as well as oral administration both have found to be very effective in protecting the animal against radiation-induced sickness, gastrointestinal and bone marrow deaths. Even in radiation-induced lipid peroxidation and elevated GSH concentration in liver, kidney, stomach and intestine, it had shown protective effect at 31st day of post-irradiation. And oral administration also protected the mice against gamma radiation induced decline in WBCs, RBCs, lymphocytes, and clonogenicity of hematopoietic progenitors, because this is very important in chemotherapy. We have seen in some of the patients that from third day of chemotherapy, there is a bone marrow suppression. WBCs, RBCs, platelets all go down. And the doctor has already fixed next cycle of chemotherapy after 15 days. But patient WBC is still at 2000, 2800. He is unfit for second course of chemotherapy. He will have to wait for 2 or 3 more weeks. In such situation, when you use Ayurvedic drugs like Bilva, subsequently or concomitantly, that will help for the patient to regain the immunomodulatory effect, to regain this bone marrow suppressed effect, and blood cells will come back to normalcy within 7 to 10 days. Patient will be ready for next cycle of chemotherapy. This is another added advantage of such products. Rohitaka is another plant. Of course, we use different plants: Amura rohitaka, Tecuma undulata, and Aphanomyxis polystachya. These are 3 different plants that we use. This is an established anti-cancer plant. You will find several articles on this. We have products also like Rohitaka arista in the market. So this is Aphanomyxis polystachya, which was evaluated for radioprotective effect. Ethyl acetate fraction was used here at a dose of 7.5 milligram per kg body weight before exposure to 1 to 5 GeV of whole body gamma radiation, which has significantly reduced the frequencies of aberrant cells and chromosomal aberrations like eccentric fragments, chromatid and chromosomal breaks, centric rings, dicentrics, exchanges, and many other chromosomal abnormalities that can happen because of radiation. Now let me quote a couple of examples of chemo and radioprotective effects of Ayurvedic polyherbal formulations. In Ayurveda, mostly we use polyherbal products. However, we do use single drugs also, but more commonly polyherbal products. This is an article published on Triphala, such a popular, such a common formulation, a combination of 3 plants, 3 fruits that we use. So this is a traditional Ayurvedic formulation being used not since centuries, but thousands of years, having immense therapeutic potential, having excellent anti-cancer activity, free radical scavenging property, wound healing property, antioxidant property, and so on. It is also revered as a chemopreventive, chemotherapeutic, and immunomodulatory and radioprotective drug. You can see the combination of 3 fruits there. On the other side, we have Terminalia bellerica, here Terminalia chebula, and of course, amla, everybody can recognize it. Accumulated evidence has revealed that Triphala modulates multiple cell signaling pathways, which may include ERK, MAPK, NF-kappa B, AKT, tubulin, P53, cyclin D1, like anti-apoptotic and pro-apoptotic proteins. It stimulates the pro-apoptotic proteins and may inhibit anti-apoptotic proteins. This is a study by Dr. Vyas, Dr. Anup Thakur, Dr. MS Baghel, and others from Jamnagar. This is a clinical study. Being a physician, I am much interested in this type of clinical evidences, which you have seen in the patients, not just limited to the laboratory. So this is, of course, a small sample study. Efficacy of Rasayana Avaleha as adjuvant to radiotherapy and chemotherapy in reducing adverse effects. So this study was carried out in a group of just 36 cancer patients who were divided randomly into group A and group B. Group A received radiation, chemotherapy, and also Rasayana Avaleha, an interventional group, and group B received only radiation and chemotherapy without Rasayana Avaleha, and they were compared. After assessing the results, it was observed that Rasayana Avaleha gave better results in controlling the adverse effects of chemotherapy and radiation in comparison with the control group. So we can definitely say that Rasayana Avaleha has a therapeutic potential as a protective drug to prevent or to manage adverse effect of radiation and also chemotherapy in those patients who are already on conventional treatment. This entire article is available online as an open access article. I'm not going for the details of this. Amrita Prasham is another drug. This, of course, being used clinically. The study published here is a preclinical study. It has shown that administration of Amrita Prasham reduced radiation-induced loss of body weight and arrested the decrease in the weight of liver, kidney, and spleen of mice. The data is here. I'm not reading out the entire data here, where it has shown the protective effect on liver, especially on GPT, serum lipid peroxide, and hepatic lipid peroxide levels. Effect starts on day 2 itself. Best result could be observed on day 7. Ashwagandha Rasayana, with respect to radiation protection, Violil and others have published an article. They said that oral feeding of Ashwagandha Rasayana to mice at 50 milligram per kg body weight for 28 days prevented radiation-induced emaciation and decrease in organ weight when compared to the irradiated controls. It also helped to increase serum GPT levels and lipid peroxidation level. Brahma Rasayana is another product which we use as a rejuvenative. This also had shown an effect of protecting animals against radiotoxicity. Chyawanprash, so popular. So many companies are advertising the products, whether it is considered as an over-the-counter drug or not. I don't want to comment on this formulation, which has got more than 56 ingredients. So if you have to prepare Chyawanprash classically without making any compromise with the quality control, it is not such a simple formulation. Just go and buy it and eat it. So there's a huge discussion that is required on this product, which is definitely having an immense therapeutic potential, not just as a rejuvenative drug. Because it is so made popular, telling that it is very rich in vitamin C. We are using amalaki here, the pulp of baked amalaki or amla. It is roasted in ghee, and during the process, vitamin C is lost. So we are not bothered about vitamin C. If vitamin C is present in Chyawanprash, that doesn't mean that it is best Chyawanprash. These are all the misleading advertisements that we are seeing all around. That's a different story. Anyway, in relation with the radioprotective activity, Chyawanprash also has got an established result against gamma radiation, which is very well published. More than 58% of survival could be established with Chyawanprash. Jageti and others have published radioprotective effect of Chyawanprash. They also found that against carbon tetrachloride induced liver damage, it has got cytoprotective effect. And a clinical study they have reported, when it was administered in a dose of 20 grams BD in BD smokers, it decreased the bronchitis which can happen because of the irritation of BD smoke. It has improved their appetite because they are all more prone to develop lung cancer, which is the #1 cancer diagnosed in males in India. Narasimha Rasayana is another product which was also found to be effective as a radioprotective in, of course, in vitro and in vivo studies. So before concluding, this is a picture even Dr. Girish Tillu had shown this today morning. Probably my next speaker will be speaking more on Rasayana drugs. They can have their therapeutic potential in managing adverse effects of chemotherapy and radiation in cancer patients, maybe by enhancing free radical scavenging activity or antioxidant effect, or by improving immunomodulation or adaptogenic effect, by potentiating antioxidant enzymes, or by reducing oxidative stress and inflammation, or by reducing lipid peroxidation, or by inhibiting mutagenesis, DNA damage, and DNA clusterogenesis. So there are many conditions where Ayurveda is handling. There is a huge list. This may be out of focus now, considering the limitation of time. I'm not going for all. There are various conditions which you can observe as a reverse effect of radiation or therapy, where we have different drugs or modalities to treat those conditions. So I have given the list of those formulations, those drugs, classical based and research based, so which are utilized in our regular clinical practice to handle such situations. Just in Triphala, we use in constipation, in pharyngitis, we use chewing of Khadiradivati. For hepatotoxicity, we use Panchakola Ghrita, or we may be using Punarnavashtaka decoction. In skin changes, we may be using simple oleation with Balataila. In cognitive deficit, we may be using Kalyanaka Ghrita, Ashwagandha like drugs, and so on. So with this, let me conclude my presentation. There are several herbs and formulations mentioned in Ayurveda which have established chemopreventive and radioprotective effects. Rasayana drugs especially have an important role to play here. Maybe free radical scavenging or antioxidant, anti-inflammatory, chemoprotective, chemopreventive, and immunomodulatory effects of Rasayana drugs will be of immense use in maintaining a healthy lifestyle and disease-free life, not just in managing cancer and its adverse effects. These are a few of the references that I had gone through while preparing this article. Thank you very much for your patient listening and also for the time given to me. Thank you very much.
Thank you, Dr. Satyanarayan. This was a very comprehensive summary of whatever is available in Ayurvedic formulation as far as radio protection and chemo prevention of cancer is concerned. The session is open for questions. Yes, ma'am, please.
Thank you so much. It's really a great learning, especially for me. I don't belong to this field. There's one very naive question. So what is the time period of effectiveness of these drugs when you take it? Like it is assumed that allopathic, another drug, they have immediate effect. So is there a differential, it's a delayed effect of them? Number one. Number two, how long you have to take it? Like if you stop, what happens?
Yes. Yes, of course, maybe this question needs much longer answer. I will try to be very specific. You may be knowing that Ayurveda is a personalized medicine, because you cannot generalize any drug or any formulation for every individual in Ayurveda. So depending upon the stage or type of the disease, and also whether the patient is exposed to any conventional treatment, whether he is already on a treatment like chemotherapy or radiation, we will have to plan the formulation, and also we can expect the result. If you are treating a particular symptom, for example, oral mucositis like symptom, or any skin changes, or peripheral neuropathy, which is very commonly seen in the case of chemotherapy, in such situations, we can definitely expect a very quick result. We not only this, we have many other herbomineral formulations also. In oral mucositis, for example, within 3 days, you can expect the results. It is published already. Comparative studies are done. Even some pharmaceutical companies also have worked on this. Now a controlled clinical trial is going on in Manipal University in the Department of Oncology in collaboration with Ayurveda on same oral mucositis. Within 3 days, you will find a remarkable relief in that. It depends upon the type of condition. So many a times we may be using these formulations for treatment of cancer itself, because I am dealing with those cancer patients for last almost 20 years now. So in various kinds of patients we do have. It may be a virgin case, stage 1 cancer, which is diagnosed by us, or it may be a terminal cancer. We feel that if you are able to bring out a smile in the face of a dying cancer, that is the greatest contribution that you can give. Quality of life, improvement of quality of life, and also survival rate is what we focus in cancer patients. Today we had an excellent presentation by a German doctor in the morning. He was talking about how excruciating it is to just dump the chemotherapy on patients in all the cases. So in many such situations, we do treat the patients even without the chemotherapy also. There are many situations where chemotherapy may not be recommended. If that is the case, so we require to administer drug for a pretty long time. I have a patient who is taking the drug for last 5 years. Patient is quite stable. Patient is, a few of them are disease free. We have patients with non-Hodgkin's lymphoma, 3 patients who are disease free for last 12 years. But still I'm giving them some rasana herbs, rejuvenating herbs. They are not doing any harm that will support to the patient. And patient feels that he is happy, he's taking something. So that is a different story. Otherwise, usually if we consider that 5 years of disease-free condition in cancer, we consider that it is almost as cured. So we need not continue the medication in such situation. Otherwise, when patient is already on disease is continuing, we cannot say that when we have to stop the medicine in many of the situations. So that may depend.
I have one question. Since this radiation always influences our immune system the most.
Yes, yes.
And what we are seeing with SARS-CoV-2 or any other viral disease, the same immune system is triggered. Yes. So what is the parallelism you found for these herbs? Are they effective in this infectious antivirals also? And if yes, then how much work is being done in India or in your institute in this area?
Yes, you are absolutely right. Because everybody was talking about immunity during COVID pandemic for last 3 years. So everybody was drinking kada as an immune booster, antiviral, anti-inflammatory, whatever it is. There are many plants here which have shown as a radioprotective. So many of them are found to be useful as immunomodulators. And some of them are excellent antivirals also. For example, I had shown Tinospora cordifolia. I have shown Phyllanthus umbilicus. Phyllanthus umbilicus is not a very potent antiviral, but maybe to some extent. Phyllanthus niruri or Bhumia malaki, an excellent antiviral. So it was studied against HPV. It was studied against even COVID virus also. Ashwagandha, Withenia somnifera, it has got a multi-dimensional application on COVID itself. So it is published. I think Dr. Girish Tilo was one of the co-authors of this. So if you Google, you will get the article on the mode of action of Withenia somnifera on COVID-19. It has got multiple actions. One among them is antiviral. Neem, for example, Nimba. Nimba is a potent antiviral. We are not using it so popularly as a chemopreventive. We use it as a radioprotective. As a chemopreventive, I have not used it much except in case of skin changes. It is very useful in the case of dermatological afflictions, whether it is because of radiation or due to any autoimmune effect. Probably we have one talk next on psoriasis. The doctor can enlighten more on that. And even as an antiviral, neem is an excellent antiviral. There are many herbs which are common as antiviral, immunomodulator. Immunomodulator need not be immunostimulant always, it may be an immunomodulation, and also a radioprotective. There are many publications are there. So personally, if madam is interested, I can share those articles also.
Hello, sir. Sir, I'm Dr. Jaswinder Singh, and thank you for this brief description of the Ayurvedic medicines that you have. I have a very small question. I want your take on modern day classification of Ayurvedic medicine. Do you feel that modern day chemistry or biochemistry should be applied on the previously known herbs to have a brief and renew classification of these medicines? What is your take on that?
The classification is something different. Of course, utilization of modern chemistry or evaluation techniques, maybe phytochemistry, will be definitely useful, because my area of interest is also the same, the standardization, quality control, all these. Classification, even in Ayurveda also, classification of herbs or plants, it is different. It varies. The classification may be morphological in some of the areas, or classification may be based on something known as a taste, which you may wonder why the plants are classified on taste. So they are having a typical therapeutic benefit on taste. We say that Madhura drugs, they are classified in the same category. There are 10 plants, say milk is a Madhura drug, asparagus racemosus is a Madhura drug. Then we have got Purulea tuberosa, it's a Madhura drug. If you see as a chemist, they are having totally different phytoconstituents, how they can be in the same category. But as you said, when we went for much more detailed studies with the help of pharmacologists, we could identify that there are some extraoral taste receptors. Taste receptors are present in the liver. Taste receptors present in the intestines. We can find bitter taste receptors somewhere in the bone. So we say that Tikta rasa dravya, bitter drug, acts on liver. So there is very interesting correlation, which I think all this modern chemistry or pharmacology will help us to understand our own classification in a better manner. The knowledge of phytoconstituent or structure of chemicals should not make our Ayurveda as disintegrated, because what happens if you are isolating an active principle and purifying and using it, then it is not my Ayurveda. It is totally different. So it can be a herbal drug, it can be some other drug, where they may be considering this, these are the anti-inflammatory reagents, these are the antioxidant reagents, or these are the analgesic potential drugs. I'm isolating and purifying it. There are a lot of issues there, because there are many other phytoconstituents which are required for the plant to show its action. So many a times, that is why when you isolate phytoconstituents, many of them do not reach to the clinical study level at all. They will fail when it comes to the clinical level. So I think whatever you said, it is a complementary, but we should, being an Ayurvedic doctor or Ayurvedic researcher, we should use this modern classification, modern analysis, maybe phytochemistry, or maybe even the pharmacogenomics aspect also, which has helped a lot to us. So that we have to understand, and we should try to correlate with the Ayurvedic basics. Ayurvedic fundamentals are very strong. If you are able to correlate and unravel those secrets with the help of these, even the modern community, a scientific community who do not understand Ayurvedic language, can be better understand this. So they can be convinced in a better manner.
So if there are no more questions, let me thank Dr. Satyanarayanan for very insightful talk. Thank you so much.
I now request Professor Kashyap to felicitate Dr. Satyanarayanan.
Hello. Good afternoon, everyone. I will talk, and next our speaker is Professor Sujata Mohanty, who is professor at Stem Cell Facility, Center of Excellence for Stem Cell Research at All India Institute of Medical Sciences in New Delhi. Her area of interest is stem cell biology, immunomodulation, exosome as cell-free therapy, and tissue engineering. She's a PhD from AIMS in 1996 and then postdoc fellowship from Mount Sinai, New York, USA. Her ICMR International Fellowship is from Kyoto University, Japan, and she's a member of Institutional Committee for Stem Cell Research for different institutes, including National Institute of Immunology, and she is a reviewer in many journals, including international and national journals, as well as in the editorial board of Journal of Repute. She published 120 research papers with an average of 8 papers per year, received 1 patent, and she filed 7 patents already. She is author of 6 book chapters. And today she is going to talk about Unani-based indigenous approaches to apprehend and abridge cancer progression. Over to you, Dr. Mohanty.
Thank you so much. I'm really glad to be here in DU campus again. I have done my graduation from DU, and it's like a homecoming here. Thank you so much, organizer, and especially Dr. Alok, for giving me this opportunity to be in the present of this August gathering. So at the onset, let me make one disclosure. Fine. I heard my previous speaker, Professor Satyanarayan, and I think there are more speakers who are going to come, and they are expert and luminaire in this field. And I am a totally novice. I don't know how come I am here. I think now I'm feeling myself a misfit. But my area of expertise is stem cells, so I would like to blend my area and a little bit touch upon why I was interested to move into Ayurveda and our conventional drug, and what is my aim. So I hope you will bear with me. And this is the institute from where I come. And in my talk, I'll be a little bit talking about the stem cells, what are the translational efforts we have made using stem cell research and stem cell technology. And of course, how we can make stem cell really more potent as an immunomodulator. Like latest COVID pandemic has shown, it's something to do with our body immunomodulation. And we were thinking that how we can modulate stem cells for its activity. There's another component of a cell, which is called exosomes. That means cells secrete out small bodies. And we were very much interested, can we leave aside the stem cells and use these exosomes for human use and diseases? And last one, I'll be talking about the organoid as a model for cancer and how we are going to utilize that. This is the latest research my lab has initiated. Let's go back into the history. So I have made my presentation keeping the budding scientists and the young faculty who are here to motivate them, and also to show them that how we at AIMS initiated this field of research. So when you see the history, so these are the 3 gentlemen who got the Nobel Prize. And in 1919, we all know Sir Donald Thomas got the Nobel Prize when he worked on mice model and gave irradiation into the marrow and tried to see that if you do bone marrow transfusion into the irradiated mice, the mice survives. So that means something there in the bone marrow, which is making these animals survive, which was the stem cell present in the bone marrow. Then Professor Yamanama, he got Nobel Prize for making your adult cells from the skin behave like embryonic stem cells. Per se, embryonic stem cells, you can only take it from the embryos. But what he did, he took biopsy from a human body, and these are the skin cells. He introduced 4 important factors so that these fibroblasts are turned into embryonic-like stem cells. The future of these iPSCs, which are called induced pluripotent stem cells, is either you can grow them in the lab, that means they will not die. If you grow your skin cells in the lab, after a few passages, they die. But these skin cells, which have been transformed by 4 factors, now it has been reduced to 3. They can grow for long period of time, you can do gene correction in them, and then you can make different type of cells using differentiation protocol, and then you can give back to the patient. So why I'm talking about gene correction, there are certain genetic mutation. Right now in India, we are using this technology to correct the factor 8 and 9, which is associated with hemophilia. So we are correcting factor 8 and 9 in these stem cells and then giving it back to the hemophilic patient. So this is one of the example. So in AIIMS also, we started our work, and I'll just give you in the highlight that whatever work which we did in the lab, how it has gone into the practices. So this is the overall scenario worldwide where people are using stem cells for the therapy. If you see here, the green area is showing the type of diseases which are recruiting patient using stem cells. These are mesenchymal stem cells. I will tell you later about them. Red means they are completed. Yellow means they are not active in recruitment, and some of them have been terminated. But this is the latest from 2015 till now. The different type of diseases you can see here, from COVID-19 to all the area, including cancers and others. In every spectrum of the disease and the disorder, these stem cells have been investigated. So this is our place at AIIMS, where we do stem cell research, both basic and clinical. We started our work in 2005 because there was some paper in 2003 which said that not only in cancer, there are other certain degenerative diseases where stem cell can be used. They were talking about two papers in Nature and Cell about heart disease. So we initiated our work, and then we moved to the phase 1 of Center of Excellence. We did multicentric trials. Then we had our basic research, increased our clinical collaborations, basic collaboration within India, outside India. Then we moved towards stem cell based product in terms of commercialization, and now having public-private partnership. So this is the overall scenario of clinical research. I will not focus on that. I'll just give you a snapshot of 2 clinical research. Just to mention it here, there are some guidelines which we have to follow, which are pertinent to India. So we follow those guidelines in our place. And all the work which we do in terms of stem cell manipulation and expansion is done in a specialized lab. These are called good manufacturing practices. So these are the few snapshots of our GMP lab. The first one is the repopulation of the eye. That means the ocular surface destruction. So if you see at the eye, you can see that it is very clear, and you can see the light is able to pass. But if there is some injury in the eye, your cornea, which is the transparent layer of the eye, it gets occluded. It is very turbid. So eyes cannot perceive the light, and the patient is virtually blind. So we try to use a special type of cells, which are called limbal epithelial stem cells. We try to isolate them, grow them in the lab, and with the aim, can we again bring back those clarity in the cornea? So I'll just show you here. This is the limbus area, which I was talking about. This limbus area, the nature has given you stem cells. So if you see the another snapshot of the limbus area, this is the limbus, this is the conjunctiva, which I call the brown area, and this is the clear cornea. So limbus area, whenever you blink your eye, few cells are lost. From the limbus area, stem cell migrate and they move over to the cornea. So we took the advantage of these cells. We took a biopsy from the limbus area, bring it back to the lab. We grew these limbal epithelial stem cells, made a sheet, and transplanted back into the patient. So again, I'll show you one of my students, Shweta, she did the basic work. We initially started with cadaveric donor. We got the limbal ring. We isolated from the ring a small piece of biopsy, which contains limbal epithelial stem cells. So this is the small piece. When you grow them in culture, you can see here the whole sheet is growing. And then we characterize them using whole panel of immunofluorescence and specific proteins. So I'll not go into the detail. We do the corneal specific markers and also the stem cell marker, which is ABCG2. When we are sure that they are maintaining their phenotype and genotype, then we take the permission from the ethics committee to do the transplantation. So you can see here, this is the normal eye. If one eye is affected, the other eye we use as a biopsy. 2 dots are made and we take around 2 millimeters of the biopsy sample from the donor eye. This work was done along with Dr. Radhika Tandon, who is a surgeon in ophthalmology. The small piece you can see here, it is cut and sent back to the lab. Then in the lab, we grow them over amniotic membrane and we give them a 37 degrees centigrade, and within 14 days, they make a whole sheet. And this sheet is given back to the surgeon. So this is the recipient eye. You can see the cornea is totally opaque here. And they first clear the surface. And then they put this sheet, which is containing limbal epithelial stem cells, and they suture it. So after some time, you can see that the vision is exactly as the previous. This is the donor eye. I want to show you this picture that taking a biopsy from the donor eye does not do any harm to that eye. This is the recipient eye, the lower panel. And after 4 years, you can see that the vision is exactly 6/6. This is a 17-year-old female with a chemical injury. This is another one, which is a 16-year-old male. And you can see here, the upper panel shows the donor eye. Lower one is the recipient eye. At 17 months, the cornea is clear. So we have done more than 1,000 cases now, and we are going to soon going to offer it as a routine therapy because it was done under a project mode. I'll just skip this one. So second one is vitiligo. So this is again a very specific problem to India because we are dark-skinned, a majority of us, and loss of pigmentation in the vitiligo causes some inhibition, especially in the younger population. So this is a case of a vitiligo. As you can see, this is loss of melanocytes, which is a pigmented cells. So the routine treatment in our aims is like you create blisters or you make skin to come up under pressure. And these are the suction cups which are placed over the thigh. And after some time, maybe after one hour, you have to cut these topmost skin layer. And this topmost skin layer is transplanted back into the vitiligo site so that normal melanocytes can repopulate that area and you get the skin coloration. So it is very painful. So that means you have to sit for some time. And when you cut these skin topmost layer, there's a scarring problem. So one of my student, Anil, he read one paper and he came to me that hair follicle contains some of the stem cells. So this is called the bulge area of the skin, the central one, and they contain stem cells and they also contain melanocytes. So that's why your hair is of particular color because of these stem cells. So then we discussed with our dermatology counterpart. We got some hair follicle cells. You can see here, these are the hair follicle cells. And under the microscope, these bulge area can give rise to small cells. These are bulge stem cells. And these cells can be differentiated into melanocytes. So lower panel is showing the differentiation of stem cells into melanocytes. So after again characterization, we went ahead with transplantation. You can see here, this was done in collaboration with our colleague, Dr. Sumesh Gupta. This is one patient, and after transplantation of these hair follicle stem cells, you can see the repigmentation. So we did it in 15 cases, and we did follow-up of approximately 4 years, and they were pretty stable. So we need to move forward in this area again, because it was showing some very promising results. So as I said that my lab is focusing on mesenchymal stem cells. So what are they? So if you see the bone marrow, majority of them are hematopoietic stem cells. So these cells are very useful for cancers or treatment of blood cancers. But there is another type of cells which is called mesenchymal stem cells. It's here. So they are the feeder cells. They give support to the hematopoietic stem cells. They are much, much less as compared to hematopoietic stem cells, but they are very beneficial in the sense every major tissue in our body has mesenchymal stem cells. So my lab, they work on placenta or the Wharton jelly derived stem cells, the bone marrow stem cells, and adipose stem cells. So the Wharton jelly, adipose, and bone marrow. So these are the 3 types of mesenchymal stem cells my lab is working on. Another advantage of these cells is, again, they can differentiate into multiple type of cells. I don't know, this picture is not coming, okay, but they are a HLA class 2 negative. That means you can easily give mesenchymal stem cell from one person to another, or you can pull together 5 individuals mesenchymal stem cells and can give without any matching. So this is one of the greatest advantage of mesenchymal stem cells, whereas hematopoietic stem cells need vigorous matching before transplantation. So this is the mode of action of mesenchymal stem cells. I will just give you a few examples in some of my slides. So these are the 4 areas which I'm going to cover in a few slides in terms of how do stem cells differentiate, what is immunomodulation, which I talked about, which is now the recent buzzword to combat major diseases. So this is the study, like what is a comparative study which we did, like how mesenchymal stem cells can do the immunomodulatory effect. Here we again took mesenchymal stem cells from adipose tissue, Wharton jelly, and bone marrow, and we saw that all 3 types of mesenchymal stem cells and their product, which is secreted in the conditioned media, can have potent immunomodulatory effect. And these are via IDO, HLA-G and PG2. And these are the molecules. I'll not go into the details. Everybody knows the native immunobiology. They are the potent molecules by which major immunomodulatory effect occurs. So if we compare 3 sources, the bone marrow, adipose, and Wharton jelly, we found the Wharton jelly, which is derived from the placenta, has the maximum immunomodulatory properties. So then we also check their functionality. So we check in the lab. So how do you check the functionality? That means you co-culture the MSC along with your patient's allogenic form peripheral blood mononuclear cells. So when we did the MLR, which is called mixed lymphocyte reaction, there also we found that it was a potent immunomodulator and it was able to decrease the proliferation of these cells, that is by the Wharton jelly. All of 3 of them showed potent immunomodulation, but again, Wharton jelly showed the maximum immunomodulation. Then we thought that, can we modulate the parent cell, that is the MSC? Can we give some signal to these MSCs so that they increase their immunomodulation? So this was our hypothesis, and there we thought of some molecules. This paper is again published. So the molecules which we chose was dexamethasone, progesterone, and interferon gamma. So because these are the molecules which are upregulated during some disease scenario, or maybe chronic or acute, so we chose these 3 molecules. So in a nutshell, I would like to say that this was the overall plan. We exposed MSCs to 3 drugs. That means DEXA, IFN, and progesterone, and we took 3 types of MSCs. We calculated the dose of these molecules and how much time do they need to do the immunomodulation, and what are the effect on, again, HLA-G, IDO, PG2, and HLA class 2. We did multiple parametric analysis, that is gene level, protein level, and also the functional assessment in terms of mixed lymphocyte reaction. So in a nutshell, I'll just say that again, there was very marked immunomodulation effect by priming these MSCs with 3 molecules, and we established different doses, and now we are going to carry forward this work in sclerosis and other immune related diseases. I'll just skip that. It is again the same with DEXA. Then there was one very important factor. Whenever we grow these MSCs, we grow under normoxia. But when you see inside the body, it is not normoxia. So I don't know why that slide is not coming. Inside our body, there's a gradation of oxygen. The bone marrow oxygen concentration is not 21%, which is the atmospheric oxygen concentration. It is around 1 to 5%. So we tried to grow these MSCs under hypoxia, and we again did this panel of tests, and we found that giving stem cells the hypoxia again increased their immunomodulation. So these are some of the papers on them. So this was our latest finding, and we are going to again do in-depth analysis that what is the mechanism by which hypoxia is increasing the immunomodulation of stem cells. Another area of research is mitochondria. So one of the important aspects of mitochondria is that it is a powerhouse. So we call it powerhouse because it produces ATP. So another important factor is that we all inherit mitochondria from our mother. So whatever mitochondria we have it inside us is from maternal transmission. So we said, okay, let's see what happens in terms of mitochondrial health of stem cells. We did a small experiment. We took epithelial cells from the skin. We destroyed its mitochondria. Then what we did, we brought together a stem cell in the close proximity of the skin cells. And we found that this MSC is making a tunnel. And through this tunnel, the mitochondria, which are healthy, present in stem cells, are being transferred to these dying cells. Because if you kill a mitochondria, cell is going to die. So this is, I'm showing you in a real time. This is the skin fibroblast. All mitochondria is damaged by rotenone, and it is stained green. Then we have MSCs. Its mitochondria is stained red. And what happens when you co-culture? You can see the green is turning into orange because some of the mitochondria is being transferred by these nanotubules. I think the picture is not very clear, but you can appreciate that this green fibroblast is turning into orange. And this is a video you can see if it runs, sorry. This was a nanotube, it is not working. And this was a small mitochondria which was traveling from the donor cells to the recipient cells. So that means stem cell can also be a good potent of mitochondria donor. And again, there are a few papers who are working on isolating mitochondria from stem cells and giving it in different diseases. Another aspect, the third and the last one, is the exosome I talked about. So MSCs, these are stem cells. They secrete some of the growth factor and cytokines, which comes into the bloodstream, and then they are distributed throughout the body. But the stem cell also secretes some of the vesicles, which are pinching from their cell membrane. And these exosomes, they contain some of the mitochondria, microRNAs, mRNAs, cytokines, and growth factors. So what is the difference between them? That they are much more sturdier. They are very, very sturdy. They are not affected by the temperature and any other adverse effect. So in our lab, we tried to see that what is the effect of these exosomes, which are derived from the stem cells, on liver injury. I don't know, the one slide is missing, so which was the overview of that. I'll just skip that. So this is the picture, like when what we did, what we created a liver fibrosis model by giving a very harmful drug. And after that, we injected these exosomes, and we found that at day 7, there was reversal of liver fibrosis in these animals. So in these mice, which were confirmed by HNE, mesenteric and Cyrus red. These are the marker of collagen deposition in the liver. I think there is some problem in the slides. Sorry for that. Some of the slides are missing. The ultimate message was there was significant decrease in the inflammation in the liver. There was decrease in the fibrosis, which was the cause of liver fibrosis, and there was regeneration of the liver. We could find some hepatocytes in the liver. This is another study I would like to highlight. Here we have tried to see if these exosomes can reverse neuroinflammation. So we have taken the example of Japanese encephalitis virus. We created that animal model and we injected exosomes into them. We found if you take JV infected rat, it dies within 3 days. But if you injected exosomes, these rats and animals, they survive. Can I have 2 minutes more? Or time is up? Okay. So this is some of the features I want to see here. That means these are the neurosphere. That means if you give JV, these neurosphere are small in size. But if you give Exosomes, the size is maintained. That means neuroinflammation is much, much reduced in them. This is again the animal studies, and this shows the survival curve of the animal. This is the mock. That means all the animals, they die. But if you take this, the green line, these are infected animals. And if you give EV, they survive. Again, by immunohistochemistry, we found that the neurons present in the brain are also very, very much healthy. These are all slides showing the regeneration in the brain also. Again, we did the immune profile of the animals also. We found that the inflammatory immune system also showed that there was enhancement of CD4 and neutrophils, and there was decrease in neuroinflammation. That means these EV can cross the blood-brain barrier and can reach into the brain. So that was the biggest finding which could find, because stem cells, they do not cross the blood-brain barrier in the brain. So this is one of the picture. The last animal is JV infected, but given exosomes, it is very healthy, and it is just jumping out of the tray. And this animal is JV infected, and it is hardly able to move. So this was the stark difference we found in these animal studies. I'll just skip these slides because of the time. I'll just go what is the way forward. This is the cancer treatment which we are going to start using the organoid model at AIIMS. So why organoid? When you use 2D system, it is different from the 3D system. We are trying to make a 3D system which can mimic the cancer. So here are some of the example of, this is a brain organoid, and this is again a brain organoid which has got the retinal cup. So retina, they are black in color. So you can see the brain organoid with the retinal cup in a dish. And these are intestinal organoids in the culture. So likewise, we are trying to make these organoid using breast cancer as a model. You can see here, these are some of in a suspension which we have made. If we take 500, the size is small. If you increase the number of cancer cells, the organoid is larger in size. These are again pictures. Some of the pictures are missing here. This is again a breast cancer organoid, and we characterize them. And now we are using different drugs to test it. Sorry about some of the pictures are not coming because it's not compatible. I made it in the Mac. So because of some publication which has come out in Ayurveda and Unani, so I was very much interested that why not I combine my knowledge of cancer stem cells using organoid model and try to test these compounds. This is just the initial studies, and hopefully we can take it forward and come out in terms of scientific validation of these, so that we can tell not only to Indian people, but also universal and global person about the effectiveness of these Ayurvedic and Unanic drug. So these are some of the drugs which we have tested, and I think I should not dwell more upon them. We have shortlisted these 4 drugs. My slides are not coming. It's coming? No, it's delay. I don't know. So we tried these 4 drugs at different combination, and we found that, I don't know. Yeah.
It's not.
It's cross. It's not taking. I think there's some compatible problem. I apologize for that. Yes. So I made it in Mac. It's not coming. I'll just tell you, like, we made these organoids, which is in a 3D structure, and we compared different drug doses on them. And there was a great reduction in the size of the organoid. And we also checked their toxicity. So they were not toxic to them as compared to the conventional paclitaxel and other drugs, which is used for the routine breast cancer. I'm extremely sorry for that. So the take-home message is that it's an evolving field. We need to do a lot of research to validate our data and then prove to other people who always see our Ayurveda and Unani in a very skeptical manner. So I think this should be the end of my slide. I would like to thank all sorts of funding and my collaborator. Extremely sorry. And thank you slide is also not coming. I thank you so much. Thank you so much. I don't know why thank you slide is also not coming. This is my team. If you can see, and if I can bring my thank you slide. This is my team. And thank you slide. Thank you so much.
Well, thank you so much for the fabulous talk. It was very informative, and I'm from the stem cell field, so it was nice to know many new things from you.
Thank you so much.
The talk is open for discussion. If there are questions, 1 or 2 questions we can take quickly.
Thank you, ma'am. For a nice talk. Ma'am, I have one doubt that because you have talked about how mesenchymal stem cells help in regeneration of damaged tissue and in immunomodulation. But if you want to target the cancer stem cell, is it that what is the main difference between the cancer stem cells and the mesenchymal stem cells? Are the mesenchymal stem cells are the part of cancer stem cells or something?
I think it's a very important question. It's also a very difficult question to answer. It's like cancer stem cells is again a very hypothesis. People are still debating, is it the cancer cells which acquire the property of stem cell, or is it some of the stem cells which are gaining the properties of cancer and becoming cancer stem cells? But we do know in a cancer, some of the major cancer cells are being killed by radiotherapy and chemotherapy, but some cells are very refractive to any therapy, and those cells are termed as cancer stem cells. So what we are trying to do in our lab are using cancer cell as a model to study cancer stem cells. And you all know that mesenchymal stem cells are also part of making cancer cells malignant cells, because they are very migratory cells. So our hypothesis is cancer stem cells are modulating the mesenchymal stem cells in the tissue, making them behave like a cancer cells, and then they get detached from the site, go into the periphery and cause secondary metastasis, or like they spread the disease. Right? I hope I answered.
Yes. Okay. Thank you. Then one question again is, it is from your exosome parts that the exosome released from the mesenchymal stem cells, and they help in the liver regeneration. So what factors actually the exosomes carries insides, so they help in
Again, a very nice question. So as I said, these exosomes are parent cell specific. So whatever factors are there in the parent, these exosomes package them inside, which include microRNA, protein, some DNA fragments. So we are working on microRNAs. So we have identified few microRNA, I didn't show you that, which are liver hepatocyte protective, which causes hepatocyte regeneration and also causes the fibrosis to reverse. So there are few microRNAs which we have identified, which were there in MSC exosomes.
Thank you. Thank you.
We'll have just 2 questions only. If you have more questions, you can get in touch with Dr. Mohanty later. Thank you.
I request Professor Chopra to felicitate ma'am.
Our next speaker is Dr. Jaswinder Singh Maras, who is an assistant professor at Department of Molecular and Cellular Medicine at ILBS. His area of interest is clinical mass spectrometry, proteomics, metabolomics, and cell biology. He is a PhD in medicine from University of Delhi in 2013. His postdoc is from Institut de Biologie, Saint-Calais, from France, and then further from ILBS from 2013 to 15. He has been trained in, as ZI as well as at Institute of Bioinformatics, Bangalore, and he has been a close friend. My junior also, we have been trained in the same lab with the same supervisor. And he's going to talk about metabolic classification of Ayurvedic compounds and their hepatoprotective effect, cellular culturomics. Dr. Jaswinder.
First of all, thank you. Thanks the organizer, Dr. Alok, for inviting me. And I also thank Manoj for this brief description. Thank you so much, Manoj. He's always kind. We have just started as an ILBS, which is a liver research institute. We have just started working on Ayurvedic compounds and Ayurvedic formulations, and now we have a project from the Ayush Ministry. So the topic which we have, so I have some preliminary data which I think will be very relevant to the Ayurvedic biologist, being a biochemist. So I will present some data on the classification of Ayurvedic compounds that we could do using modern day biology, particularly metabolomics and proteomics that we could do in these cells. So the topic is metabolic classification of Ayurvedic compound, and we how do we go about it? So Ayurveda is all know that it is the medicine for India. I mean, we have done and we are utilizing it, but modern day classification of such old medicine is needed, and everybody needs to know what are the molecular classification for a particular Ayurvedic component. So using the omics speciality, what we can do. So what we have done is we have tried to search certain metabolites and certain proteins that we could harbor in these Ayurvedic formulation, which may be giving you the medicinal effect. So we have tried to classify these compounds. So these are the proposed drugs we are going to work on, and most of them are very well known to you. For example, Guduchi, Amlaki, Rohitika, and Triphala, and Nimba, and other formulations are also, for example, Arogyavardhini. So we intend to work on them. So going forward, the idea and the aims of the work is to first characterize the heavy metal profile in various Ayurvedic formulations. We wish to do multi-omic classification of Ayurvedic formulation, as well as the plasma samples of patients who are undergoing Ayurvedic formulations or taking Ayurvedic formulation, with the effect those who get benefits and those do not get benefit. What are the multi-omics effect of that? We wish to correlate the heavy metal profile with the multi-omics profile and the clinical profile for estimation of the hepatoprotective or hepatotoxic effect. So whether there are hepatoprotective or hepatotoxic effect. And further, this is the very important point which we think is that chemical modulation of the hepatotoxic compound. So if we are able to identify or accurately characterize the Ayurvedic formulations or Ayurvedic drugs, we may be able to propose a revamp of the Ayurvedic medicine. So this is what we intend for. So we have this tie-up with the All India Institute of Ayurveda, and we collected the Ayurvedic compounds from the All India Institute, which they are prescribing every day. And we also use some commonly used Ayurvedic drug, for example, Giloy, Shilajit, and other. In this particular work, we have compared to these things. For example, we worked on the metabolomic classification of Guduchi, Giloy, which is again the Guduchi, but in the tablet form, the stem form, the tablet form. We worked on Shilajit, and we worked on the Arogyavardhini Gutika. So the Arogyavardhini Gutika is a formulation which also contains Shilajit. Therefore, the chemical classification was found to be similar. We'll see. So the first is the Giloy. It is very well known herb. Everybody from last 2 years may be having this because of its antiviral effect, and it has been very popular nowadays. And it has an anti-diabetic, anti-tumor effect, and also known to reduce the AST, ALT levels. So it has an hepatoprotective effect. So the key known compounds which are known are alkaloids, dibretanoids, glycosides, and others, aliphatic and steroid. But what are them? What are these alkaloids? What percentage does they contribute to the Giloy is not known. Which of them has the highest percentage? What is the effect if Giloy has an antipyretic effect or antiviral effect? What is that compound that is contributing majorly to the antipyretic effect is not known. We're giving Giloy. So there are many papers who people have worked on, and they have tried to classify these compounds. Again, Shilajit, which is very well known for its immunomodulatory, anti-inflammatory effect, and also it can work on HIV, AIDS, other things. But again, the molecular classification of Shilajit is also not known. So the key compounds in the Shilajit is particular heavy metal. So these are the heavy metal compounds. And if you see, there are two types of Shilajit, the Afghani Shilajit and the Pakistani Shilajit. And then if you see, the amount of iron in them is very, very different. So all the metal contributes to various biology, and being working on the iron biology for many years, you can see that the Afghani Shilajit, maybe it is working better than the Pakistani Shilajit or whatever that means. So one has to have a clear classification of the Ayurvedic compound in the modern terminology. So being very new to this Ayurvedic biology, what we have done is we have adopted the omics technology to search what are the metabolites, what are the proteins that are there in these molecules. Okay. The other part is this Arogya Vardhani, which is having Shilajit as such, and these are the amount. But this is the only classification of the compound that is given. If you see, for example, if you take Guduchi tablet from Dabur, it does not give you what compound it has. It says Guduchi, that's it. It does not tell what all compound it has, what percentage it has, because there is no classification of that. So for that purpose, we started this work. So what we have done here in our work, it's a very small effort which we have started. So what we have done is we have taken all the compounds, and we have using the published technologies that we have done metabolomics, lipidomics, and proteomics of these compounds. So all these compounds in the form of tablet or in the form of stems were crushed, they were sonicated, and then the supernatant or the crushed part were used to isolate metabolites, the lipid forms and the protein forms. So to search, it is very important, being a metabolomics expert here, it is very important that there should be a database. So there is no known database. So we have utilized the plant metabolome database, which gives us an idea of the alkaloids. There are two types of this, the secondary metabolites and hormones. Then the secondary metabolites are divided into alkaloids, which are different. There are many alkaloids, then phenolics, which are very how many, and then terpenoids and other metabolites, and then there are many hormones. So when we annotated, when we worked on these compounds, we were able to identify 1020 compounds in the mixtures. And if you see that these are the subclasses and the classification of the compound, how many metabolites belong to which subclasses are here. For example, indole alkaloid, 126 metabolites which were of the indole alkaloids were found in different compounds. Let us see what we found. So whatever people found, whatever is known, for example, berberine is very well known to be high amount in the Guduchi. So we found berberine to be very much high in the Guduchi and Giloy tablets. That is what. So, and other like fulvic acid and humic acid are known to be part of the Shilajit, and we found them to be very, very high in Shilajit and as well as in the Arogya Vardhani good Vatika. Then we did the global metabolomics for Giloy tablet as well as the stem, and we compared whether the tablet and the stem is different, and can there be difference in the tablet, which is nothing but a mixture of grinded powder of stem given in a tablet and a stem which is not grinded, but you are freshly grinding it. So is there a difference? That the metabolomic profile shows little bit difference. So the indole alkaloids in the Giloy tablets were higher, but the alkaloids were 60%, 61% in the Giloy tablet versus Guduchi is 57%. And if you see, the indole alkaloids is 36% in the Giloy tablet, and here only 15%. Let me go to the other part. If we compare the Arogya Vardhani to the Shilajit, so these as Shilajit is a part of Arogya Vardhani, the comparative profile was similar. For example, the Arogya Vardhani has 55% alkaloids, and the alkaloids in Shilajit was 53%. and the other, for example, abscisic acid and other compounds were also similar. For example, 4.3, 4.7% here and 6% here. So if we see what are the alkaloids, what kind of alkaloids and what is the percentage of alkaloid. So the betaine alkaloids were higher in the guduchi. In the guduchi is basically the stem, and giloy means the tablet form. So was found to be higher in the guduchi stem, and indole alkaloids were higher in the giloy tablets. So peptide alkaloids were higher in shilajit. The arachidone alkaloids were higher in guduchi. So these alkaloid was higher in the Arogyavardhani, and these alkaloid, they were different alkaloids for which were dipeptoid alkaloids were higher in the guduchi. So if you see, there are different type of alkaloids, and the percentage varies for if you compare the stem as well as if you compare the tablet. So we found. So what are the key markers of the alkaloids? What alkaloids we have and how they are? So for example, this is a very, very important estimation where we saw that in terms of conicin, this is conicin, which was found to be very much high in giloy tablet, which is 10% and 11%. Conicin is known for its antipyretic effect. So conicin could be one of the compound why having giloy you have a decrease of fever. So therefore the percentage amount of conicin in giloy was around 10% to 11%, which was not there in Arogyavardhani Gutika or Shilajit. Further, there are many compounds which we found and were also published, and people were also showing the effect of that. For example, one of them is conicin. If you see phenolics, then there were many phenolics which were different, like minor flavonoids, benzofurons, flavones, and you can see the different effects. For example, the flavones were very, very higher in Arogyavardhani Gutika, and quinones were very, very higher in the Guduchi as well as Shilajit, and tannins were higher in Shilajit and isoflavonoids. So we have a brief description of what are the key phenolics of these Ayurvedic drugs. So these are the key phenolics. The darker the color, the more percentage that compound is present in these comparisons. So you can definitely see that which compound has different concentrations in the sample. Again, we searched whether we have something which is published or are we getting something which is known, and we found that yes, there are published. For example, this plugman is published and it is known. Then we went and searched for terpenoids, the diterpenoids, the monoterpenoids, the triterpenoids, the phytosterols, the carotenoids, and the other terpenoids and steroids. These are steroid saponins. They were differentially present in the chemical classification is different. So the key compounds that they were there, for example, these steroids were 26% in the Arogyavardhani and 44% in this was 44% in the giloy tablet. Again, we searched whether some of them have been known to be published and have an clinical approach, so we could find certain of the metabolites. Then we searched for the hormones. The next factor is the hormones, and we found that the gibberellins and the abscisic acid and auxins and ethylenes were all different. The cytokinins were all different, and you can find different concentration of these cytokinins. So again we searched for other non-hormonal molecules which we could found, and we can see that the hormonal profile were also different for different Ayurvedic compositions. Further, abscisic acid was known for to be used to combat the pre-diabetic and metabolic syndrome. So other molecules which are there other than the other hormones which are significant were these other markers which were significantly present. For example, we could find some nicotine, we could find some carine, D-glucose. So these are all present in the molecule. So to conclude, the first part of our work is that we tried to do the metabolic classification of these Ayurvedic medicines, and we could find that the Arogyavardhani Gutika compared to Giloy in terms of tablet or Shilajit, the secondary metabolite composition has 89% secondary metabolite, and the hormones were 10%, contributing to 100% of the aerogravity. And if you see Giloy and Guducci, they were kind of similar because they are same, and they were giving similar profile. And these are the known or significant molecules which we could identify. We could pick one or two of these molecules now, and the next is to identify those molecules which are present in the higher concentration in these Ayurvedic compound, and go for chemical purification of these compounds, and thereby use them to see whether they are hepatoprotective or hepatotoxic, and compare them to the native compound, whether the effect of that compound is more than the native compound. Because the very important part is that Ayurvedic medicine is very good, but prolonged use of Ayurvedic medicine also causes problem. And therefore it is very important to identify what is the component that has the maximum utility in that Ayurveda. And if we could identify those components, we can make an allo Ayurvedic medicine which would be used for everybody. And for example, just for an example, which we are thinking, that if we go and see conazin is known for its antipyretic effect. If we take out and purify conazin and make a tablet out of conazin and put conazin in front of crocin, you will get that a conazin will have more effect than crocin, and it would be non-hepatotoxic. So crocin, which is an hepatotoxic compound, maybe Ayurvedic manipulations could lead us to get better medicines which will have the non-hepatotoxic effect. So the project is continuing. We are very initial in the project, and I would like to acknowledge my boss, Professor Sareen, and my team and ILBS, and I'm ready for your questions. Thank you so much.
Thank you, Josie. It was a nice talk, especially when we know that in metabolomics, there is no standard database. Proteomics and lipidomics still, proteomics standard database are there, but not in metabolomics. So very nice work. The talk is open for discussion. We can take some questions quickly. Yes.
Jaswinder, nice presentation. And also, like recently, there are many reports about Giloy and its hepatotoxicity. And there was an article in Journal of Clinical and
that was retracted also for some reason. And then people are saying that Giloy has hepatotoxicity. See, there is. And the idea people are giving is, the Ayurvedic guys are giving. I'm not an Ayurvedic guy, neither I'm a allopathic guy. I'm a biochemist, which is trying to see Ayurvedic medicine and see what is there in it. So what Ayurvedic guys are saying, that the species of Giloy that they are taking is different. So they are making it and the species, but I don't feel that the species is different, is that the chemical composition is different. So it is better to first classify the chemical composition and see what are the compounds that are present in that. So at least I would be very happy, at least if our efforts in this coming 3 years makes Giloy tablets having a detailed list of all these compounds, saying that the percentage of that compound is this much, and this is the accurate formulation for Giloy. So the Giloy should be tested for these compounds. Yes, it qualifies and then can be taken, rather than just taking it wherever and whosoever is getting. So this is what we think. I mean, as a biochemist, I think.
And also, like, there is a possibility of fungal contamination,
Sure.
aflatoxin contamination.
Sure. There are. So therefore, as Manoj said, first, metabolomics, the databases are not available. The plant database of metabolomics is very difficult. And the other part is that we have to search it in multiple databases to see whether we have fungal metabolite, we have bacterial metabolite. For example, Shilajit is actually moss, which is growing, and because of the bacterial and fungal. So one has to characterize whether that is a fungal concoction or that is a bacterial concoction that is working. We don't know, we're just having it. We are just taking it, it's an immunomodulator. Okay, let's eat it. But what is it is actually needed. And we have started, I don't know how much we can go forward, but this is just a neonate step towards the actually what we need to do.
Okay, thank you.
Thank you. Well, not how much, you need to keep going because that's important. And it's always a question that how much is too much. So we need to figure it out.
Thank you so much. Thank you. Very interesting and I think very fruitful work. We are looking forward to your work and outcome. So very small query. You are trying to isolate different metabolites from them and trying to see if one metabolite can replace a conventional drug. Whatever I understood. So I was just wondering, is it a single drug which is working or is it a combination of everything which is coming? If we take one drug, it might fail in its action. So
it's a systemic effect. Everybody says it's a systemic effect and everybody knows it's a systemic effect. Now, one thing is, if it is a systemic effect, that means that some compounds are targeting some things and some compounds are combining and targeting the other. For example, the immunoreactivity can be, down regulated by combination of certain compounds, but we should know the combination. It is very important to know the combination. It's not that, okay, it's a systemic effect, so let's not study the. But if we characterize A to Z of that compound, then we will be able to know that A to F is immunomodulatory, F to Y is antipyretic, and then Z is actually doing nothing. So why to have Z? Maybe Z is having an hepatotoxic effect, prolonged use of Z. So just take out Z of that. That is revamping the Ayurvedic formulation. This is what we could understand.
No, it's very true because it's combined effect. Whenever you are actually tweaking the backbone of any natural product, it's most of the time it's losing its impact. So yes. Any more questions there? Well, look like we don't have any more questions. Thank you, Jaswinder. Thank you for a wonderful talk. Thank you so much.
I request Professor Chopra to please felicitate Dr. Jaswinder. I would like now to thank both the chairpersons. I request Professor Chaturvedi to please felicitate Professor Chopra and Dr. Kashyap. So after this session, we actually have another presentation, which is by Professor Mehdi Shakibai. He is a professor at Institute of Anatomy, University, Munich, Germany, and his area of interest is in inflammation and tumor biology. Professor Mehdi did his PhD from Free University, Berlin. He's also was a professor at the Free University of Berlin, and we'll now listen to his recorded seminar. So I request the audience to stick around. Thank you.
he is actually I'm very sorry. I can Munich, here is Shaki Bhai. I'm very sorry, I cannot be physically there, but I'm very happy to show you some part of my result by this way. First of all, I would like to thank the whole organizing committee for this very well organized Congress, particular Dr. Alok. The topic of my talk today is multifunctionality of collagen A in inflammation, chronic diseases, and cancer. Inflammation, chronic diseases, and cancer. We know today there is almost no disease that is not directly or indirectly related to inflammation. In other words, there is no way around it in medicine. The main sources of inflammation cause, they are food factors, ultraviolet radiation, environmental pollutants, alcohol abuse, nicotine, microorganisms like bacteria and viruses, stress. They cause inflammation. And this inflammation could cause in every organ itis. We know today every organ can get itis. And when these inflammation get dysregulated and can get a chronic process, they can develop cancer. Even Rudolf Virchow in 18th century talked about inflammation and its association with atherosclerosis, rheumatoid arthritis, multiple sclerosis, asthma, Alzheimer's disease, and cancers. The role of genes environment in the development of cancer, we know today even less than 5% of all cancer cases are caused by genetic defects, and 90 to 95, 98% are caused by environment and lifestyle. It means this is a great opportunity for cancer prevention. As you see here, among these factors, diet may be the most important environmental factor. Dysregulated chronic inflammation caused by lifestyle factors causes chronic diseases, including cancer. Regarding pathogenesis of cancer, this is genetics and epigenetics. Genetics, it means expression of a genetic disposition or instability, damage of the genetic materials or errors in the reading of the genetic informations. Regarding of epigenetics, this says mechanisms that exert a heritable and indirect influence of gene expression without permanently change the DNA sequence. In contrast to genetic alterations, epigenetic changes occur much earlier during cancer initiation and progression. They are potentially reversible, are easily influenced by environmental and dietary factors like meat, fat, smoking, etc. With other words, epigenetic changes due to dietary polyphenols are a new medical approach. We know today the very famous pro-inflammatory transcription factor NF-kappa B is linked to inflammation. That is a pivotal transcription factor in chronic inflammation disease, and we know that NF-kappa B does play a main role in cancer development and progression. Central research principle for most type of inflammation, it means pro-inflammatory environment cause activation, phosphorylation of a lot of transcription factor and signaling molecule, including NF-kappa B, and they cause inflammation. NF-kappa B, we now today regulate at least 500 different genes and genes proteins, which are involved in metastasis, inflammation processes, anti-apoptosis, pro-apoptosis, transcription factors, stress response, proliferation, and cytokines processes. With other words, NF-kappa B is a powerful pro-inflammatory cytokine. Cancer treatment requires suppression of multiple cell signaling survival pathways, inhibition of multiple cellular survival signaling pathways in tumor cells by non-toxic multi-target drugs is essential. For that reason, natural polyphenols are known to exhibit a wide range of anti-tumor, anti-oxidative, anti-inflammatory effects as free radical scavenging, inhibition, or induction of apoptosis. Cancer is one of the most commonly diagnosed diseases, and its associated mortality is a very significant health problem worldwide. Despite numerous recent improvements in diagnosis and monitoring, the overall survival rate for cancer has not improved. In fact, nearly 50% of cancer patients develop chemoresistance and recurrence, suggesting that available therapies are unable to control this deadly disease. That is the major cause of failure of cancer chemotherapy. With other words, activation of multiple cellular survival signaling pathways, which are activated by tumor cells, is the primary cause of chemoresistance and tumor cell recurrence compared to conventional mono-target therapies. Why is NF-kappa B a good target for development of cancer drugs? Most cancerogens activate NF-kappa B. Tumor cells often produce an activated form of NF-kappa B. Even tumor microenvironment can trigger NF-kappa B activation. NF-kappa B activation blocks apoptosis and mediates tumor cell proliferation. NF-kappa B activation leads to resistance to chemotherapeutic agent and gamma radiation. Several genes involved in tumor initiation, promotion, and metastasis are regulated by NF-kappa B. With other words, inhibition of NF-kappa B activation suppresses cancerogenesis. Classical tumor treatments, the mono-target therapies, are actually unsafe, ineffective, high recurrence prevalence, and very expensive. Tumor cells are extraordinarily intelligent cells. They develop differential resistance pathway. For that reason, we call that multi-drug resistance. They act on more than one mechanism at the same time. And monotarget chemotherapy treatment, you can imagine, cannot fight at the same time against different signaling pathway which are involved in apoptosis, proliferation, recurrence, and metastasis. For that reason, phytopharmaceuticals that modulate multiple cellular targets, such as anti-inflammatory, restrict energy, creating an alkaline environment, promote apoptosis, prevent proliferation, migration, invasion, and therefore they are effective chemosensitizers for tumor cells against conventional therapeutics. Today, I'm talking about Calibin A and cancer. Calibin A, its structure from turmeric, is a new curcuminoid isolated from turmeric. Like curcumin, Calibin A can inhibit the tumor cell growth and induce apoptosis in drug-resistant human cancer cells. Last month, we could publish a paper, multifunctionality of Calibin A in inflammation, chronic disease, and cancer. As you see here, we summarized here all transcription factors and signaling pathway which are involved in inflammation and in tumor development are blocked by Calibin A. And Calibin A at the same time is able to inhibit, stimulate caspases. In other papers, we could show Calibin A suppresses NF-kappa B and NF-kappa mediated proliferation, invasion, and metastasis in CRC cells induced by TNF-beta. As you see here very well, in 3 different CRC cells, Calibin A inhibits the proliferation of cells, and even when the cells are stimulated by TNF-beta, are inhibited significantly their proliferation. We can see here very well, Calibin A is able to inhibit significantly the phosphorylation activation from NF-kappa B. Even the cells are activated by TNF-beta, phosphorylation and activation of NF-kappa B is significantly blocked. Even NF-kappa B mediated proteins which are involved in proliferation, invasion, metastasis, like integrin beta-1, CXCR4, MMP9, T67 are blocked. And at the same time, caspase-3 is involved in 3 different CRC cells. And at the same time, when the cells are stimulated by pro-inflammatory cytokines TNF-beta, Calibin A inhibit the mentioned proteins and activate again caspase-3. With other words, NF-kappa B seems to be one of the major target transcription factor for Calibin A. Even as you see here very nicely, Calibin A blocked the interaction between phosphorylated NF-kappa B and DNA. Next paper, we could show more details about the role of NF-kappa B and Calibin A interaction. As you see here, we use in this paper BMS that is very specific EKK inhibitor. We wanted to compare the function of calibin A with BMC. As you see here very well, the phosphorylation activation of NF-kappa B is dose dependently inhibited by calibin A exactly in the same row like BMS. Even the proteins, they are regulated by NF-kappa B and involved in migration, metastasis, and proliferation, cyclin D1 and MP9, they are inhibited. In contrast to that, caspase-3 is activated in the same row like BMS. In this picture, you see here very well the expression of anti-apoptotic genes are inhibited by calibin A in the same row by like BMS, serving VCL2 and VCLXL. Next paper, we could show calibin A potentiates the effect of 5-FU and TNF-beta against human CRC cells and the role of NF-kappa B. As you see here very well, TNF-beta stimulate in this same row in normal CRC cells and even in CRC resistant cells. However, in the presence of 5-FU in normal CRC cells, the proliferation is inhibited, but not in resistant cells. However, the TNF-beta stimulated cells in normal CRC and in resistant cells in the presence of 5-FU and calibin A is in the both cells is significantly inhibited. This potentiation between calibin A and 5-FU you see here in regarding of migration. You see here in normal CRC cells as well as in resistant cells, the proliferation, the migration is significantly inhibited. Interestingly, in the presence of 5-FU, the phosphorylation of NF-kappa B is activated dose-dependently in both of cells. However, in the presence of calibin A and in the presence of TNF-beta, the activation of phosphorylation NF-kappa B is blocked very significantly. Against, we see here potentiation as a synergistic effect between calibin A and 5-FU. And even in the regarding of expression, the proteins which are activated by NF-kappa B and involved in migration, proliferation, and metastasis like beta-1 integrin, MMP9, CXR4, Ki67 are activated by 5-FU. And as you see here, caspase-3 is inhibited by 5-FU alone. But in the presence of calibin A and TNF-beta, the mentioned proteins are inhibited and caspase-3 is very well activated. The next paper, we could show the EMT and CRC in combination with calibin A treatment. We have used in this paper cytochalazin D. As you see, cytochalazin D inhibits cytoskeleton proteins in the cells. We wanted to show calibin A could inhibit cytoskeleton. And as you see here, regarding of migration, dose-dependently, calibin A inhibit migration of the cells like cytochalazin D in both kinds of cells. And cytochalazin D is able to inhibit the migration of cells. And we could show here in the same row, calibin A inhibits phosphorylation of NF-kappa B like cytochalazin D in both kinds of cells. Phosphorylation activation of FAK, focal adhesion kinase, which is important for polymerization of cytoskeleton proteins, is inhibited by calibin A, like cytochalazin D, And even the activation of caspase-3 is promoted by calibin A, like cytochalasin D in both kind of cells. With other words, it seems that cytoskeleton proteins, they are the next target proteins for calibin A. We wanted to see the functional cooperation between phosphorylated NF-kappa B and SLAC. SLAC, we know that is very important transcription factor regarding of AMT. And as you see here very well, immunoprecipitated NF-kappa B are linked with SLAC. And calibin A, like cytochalasin D, are able to inhibit these links. With other words, SLAC is the next targeting molecule for calibin A, and vice versa. When we immunoprecipitate with SLAC and immunoblot with phosphorylated NF-kappa B, we see the same results. The next paper we could show here, even cancer stem cells are the main target of calibin A. As you see here, stem cell marker ALDH1, CD44, CD133 are downregulated by calibin A in the presence of TNF-beta, dose dependently. Calibin A has an anti-inflammatory effect through different signaling pathway, as summarized here. And calibin A as anti-cancer agent is able to inhibit different cancer cells, for example, breast cancer cells, CRC, as I showed you, gastric cancer, head and neck cancer, hepatic cancer, leukemia, lung cancer, lymphoma, and multiple myeloma. I summarize here again molecular regulatory targets of calibin A regarding of kinases, transcription factors, apoptosis, cancer stem cells biomarker, proliferation, metastasis, EMT biomarker. My last slide, I summarize again, calibin A is able from outside to inhibit, block, even receptor-ligand binding, is able to block phosphorylation of NF-kappa B, translocation from NF-kappa B in the nucleus, and even to block the proteins which are involved in inflammatory invasion, metastasis, chemoresistance, and angiogenesis processes. Conclusion: to prevent tumor cells resistance to conventional mono-target chemotherapeutic agents and to chemosensitize tumor cells through chemotherapeutic agents is the initiation of multiple cellular survival signaling pathway in tumor cells by non-toxic multi-target drugs is essential. Cancer treatment requires suppression of multiple signaling and survival pathways. I thank you for your attention. And if you have a question, I would be very glad and happy to get from you by mails your question. And I wish you very nice Congress. Bye-bye.
इंस्ट्रूमेंटल 2 मिनट लगा दो। We will be starting the next session shortly. We will be starting the next session shortly.
ये कलम बनारस इंक से है। ये अंग जयपुर अंग है। ये पटियाला अंग है, पंजाब। बिडार अंग है।
Welcome back, ladies and gentlemen. We have gathered here now for public lecture 3. Chairpersons for this session are Professor Madan Mohan Chaturvedi and Professor Kishore Singh. At the last moment, our pre-decided chairperson, Dr. Avnil Mishra, could not join us because he is stuck in Haldwani. And therefore, Professor Kishore Singh will be co-chairing this session with Professor Chaturvedi. Professor Madan Mohan Chaturvedi is advisor cum provost at SGT University, Gurugram, Haryana, India. His areas of interest are chromatin remodeling and regulation of gene expression, mechanism of cytokine action and signal transduction, inflammation and cancer, epigenetic regulation during liver regeneration. Professor Chaturvedi has a PhD from Department of Zoology, Banaras Hindu University. He was the head and professor at Department of Zoology, University of Delhi, and Dean Research Life Sciences, Department of Zoology, University of Delhi. Professor Chaturvedi was a visiting professor at MD Anderson Cancer Center, Texas, USA. He was also elected as president of Indian Association of Teachers in Biological Sciences for the year 2016 onwards. Professor Chaturvedi was named Man of the Year, American Biographical Society, Raleigh, North Carolina, USA. In 1988, he received the Indian National Science Academy Young Scientist Medal. In 2017, he was the external member of Board of Research Studies at Northeastern Hill University, Shillong, Meghalaya. Professor Chaturvedi was awarded the Swiss National Science Foundation Fellowship from the University of Zurich, Switzerland in the year 1986. I welcome you, sir, for chairing this session. Our next chairperson is Dr. Kishore Singh. He is the director, professor at Maulana Azad Medical College and director of Delhi State Cancer Institute. He is also editor-in-chief of Journal of Cancer, Research and Therapeutics, and Radiation Oncology. With this brief introduction, I hand over the session to you, sir, to proceed with the public lecture proceedings. Thank you.
With the permission from my co-chair, Professor Kishoreji, it's my privilege to invite you all to the last session of the 3 days academic feast, I would call it, as which you all have witnessed. But this session is an equal and very important session. Now, as you all know, this session is going to be delivered by none other than, if I take the liberty to call him the Bhishma Pitamaha of TNF and the nutraceuticals. You all have been listening from day one that he entered into the domain when none even thought of. I mean, I will also share some of my personal, give me 2-3 minutes time, because my association, Professor Agarwal, had been very long. And so he started working in the field when no one even thought of, and when we started working, there were a lot of comments, a lot of ridiculing that what are you going to do, what you are going to achieve. But with his commitment and passion, he kept on working. And now the work started with curcumin way back in 1993. And now you all know the rest of the history, the rest of the world has followed it with some or other. And then after this curcumin, there was never looking back. The number of phytochemicals, number of compounds, number of spices, number of plants, establishing the concepts, establishing the proof, and that he entered into to show to the world that there can be an alternative therapy. And maybe you listened from him that there can be, instead of a farmer's market, what he calls PHARMER, pharmacy market, to the farmer's market, FARMER, farmer's market. Now, to those who still do not know him, let me just tell you that this is an old thing, don't read it. His total publication is more than 900. Citations are more than 1,70,000 something. H index is 211. I index is 622. And twice he has been awarded the top 100 best, most highly cited scientists in the world, not only once, twice. And rarely it happens, the people from the industry, academia moves to the industry to the academia. A lot of people go from academia to industry. So having worked as one of the 10 founder person of TA and Genentech, later in '89, he moved as a full professor grant to MD Anderson Cancer Center. And at that time, again, let me tell you, the term biological therapy had just emerged. The Genentech TNF was tested in that lab and that department, which was chaired by Jordan Gutterman, and not TNF, interferon. Interferon was first tested as a therapeutic molecule, and consequently the department was named as a biological therapy first in the world of kind. And Professor Agrawal was professor invited from Genentech to be a professor at that department. And then he has a long journey at MD Anderson Cancer Center, trained more than, the list is long, 100 postdocs and other. And he established not only a school within the US, across the globe. The list is on and on. I can keep telling across the globe because I've been associated with, since 1993, I got a chance to visit him. And since then, my association has still been continuing. So with these few words, I will not take long because it's a last session and all of you want to get concluded. I take the privilege to invite you, sir, to give the public lecture. Professor Agrawal, please.
Dear friends, ladies and gentlemen, first of all, I would like to thank the organizers for giving me this opportunity to be here with you. And I'm very happy to be here. And all of you know who the organizers are. And that is Professor Alok Bharati and Anju Shrivastava. So if it was not for them, I will not be here. Second, I would like to thank Dr. Madan Chaturvedi for his very generous introduction this evening. And it is very nice of him. It is very, very flattering. And last of all, I would like to thank all of you for being here. If it was not for you guys, I will not be here. So thank you. And today, this is my first slide. So today, as you can see, what I'm going to talk about, targeting inflammatory pathways for prevention and treatment of cancer and other chronic diseases by agents derived from Ayurveda. And as all of you know, India is a land of Ayurveda, and it is the oldest science known to man. And it is also, India is also land of spices. And people have been looking for spices for hundreds and hundreds of years. And as all of you know, that Vasco da Gama and Christopher Columbus and Jose Cabarrillo, and all these three guys were looking for spices. And they knew that spices are connected with India. So they apparently found some spices in America and they thought that those were Indians, and those turned out to be now called American Indians. And so it is a Vasco da Gama who actually discovered the spices in India. And we organized our first Nutra conference. Actually, it was second one is in Goa, and that's where Vasco da Gama is from. So, and then, so you can see Ayurveda, spices, and other nutraceutical. So I'm kind of going to give you a kind of overview how we look at all of this, and apparently that plays a very important role in various diseases known to man. So with that, that I already mentioned, that thank to Alok Bharati and Anju Shrivastava for organizing this conference, and they have done an excellent job, and let's hope that this thing continues. And this is just a flyer. And we had formed another society called International Society of Translational Cancer Research. And as you can see, that the first conference we held was 2006 in Trivandrum, and the last one was in Varanasi in 2020. And so this also lasted very well. And it just so happened to be that I've been working on cancer for last 40 years. And some of you may know who Steve Jobs is. And Steve Jobs happened to have a friend. His name is Bob Swanson. So both the guys moved to a Bay Area, San Francisco area, and they both started a company. Steve Jobs started a company called Apple, and Bob Swanson, his colleague, started a company called Genentech. So, and Genentech was the first biotechnology company ever. And so I got hired by Genentech, and that is where Bob Swanson came to me and he said, Bart. I said, wait a minute, my name is Bharat. He said, can I call you Bart? So Bart in San Francisco means Bay Area Rapid Transit. So that was the first time I was given a name Bart. And so all the white people, American people, they cannot pronounce Bharat, so they call me Bart. So that is where I started my career on cancer. And I've been working on cancer since 1980. So it is now more than 40 years. And these are the conferences that we were organizing on cancer. And then we also asked that it is the mother that brings us in this world, and Mother Nature is there to take care of us. So the question is, how does Mother Nature takes care of us? And that is where the word nutraceutical came along. So you have a pharmaceutical. Pharmaceutical are the drugs that are made by man, whereas nutraceutical are the drugs made by a Mother Nature. So these are natural. So that is where we found another society. As you can see, the first conference we held was in Cochin in 2016, and then in 17 we had in Goa, and then Dehradun, and then Guwahati, and now in Delhi. And then you can see some of the others that we have planned in the future. So this is again making people realize that what, as I mentioned before, that Mother is the one who brings us in this world, and Mother Nature is there to take care of it. And the question is, what is there in the Mother Nature that takes care of us? And that is what this conference is all about, this nutraceutical conference. So these are some of the organizers of the nutraceutical conference. I just put it down there, the pictures, and these WhatsApp has become Zoom has become the boom, and so we Zoom every now and then and meet, and these are the people that we are meeting through Zoom and to talk about nutraceutical. So as I mentioned that I got into cancer, and the way I got into cancer was year 1980. I already mentioned you Bob Swanson and Steve Jobs. They both were together, and one started Apple, other started Genentech, and it is Bob Swanson from Genentech, the first guy who got me into cancer research. And that was year 1980. So when he asked me that, okay, he wants me to find a cure to cancer, and I did not know anything about cancer, and there was nothing known about the immune system. So it is at that time we started grinding up the immune cells and isolating factors that will kill only cancer guy and not the normal guy. And so that is where we identifying the first factor, and that is called tumor necrosis factor. We were the first one to purify, sequence, and clone. And it just so happened that my colleagues, they were the first one to clone interferon, and they were calling it interferon alpha, beta, gamma. So when I asked TNF that where the TNF is coming from, so it was coming from the immune system, and the question is which cells in the immune system, and it turned out to be, it was coming from macrophages. And as you know, that the immune system has macrophages, and that also has lymphocytes. And from lymphocytes, we identified another factor that will kill the cancer guy and not the normal guy. So we did not know what to call it. So we started calling it TNF beta, and originally it was called lymphotoxin. So lymphotoxin became TNF beta. So a lot of stuff that I'm going to tell you today is either done by TNF alpha or TNF beta. So this is just to give you a kind of a summary about some of the paper that we have published on TNF alpha and TNF beta, and it is all out there. Any of you can look it up, but here I just put it up in one of the slides. And by the way, if anybody is interested in any of the slides, it is available to you and just for general information. So we have a reviews like signaling pathways of the TNF superfamily, a double-edged sword. What does that mean? That one of the edge of the sword is good for you, another edge of the sword for bad for you. It is made by the immune system. Or historical perspectives on TNF and its superfamily. And again, that is all published, but it is just trying to give you that overview. And so it turned out that all of you heard of probably MD Anderson Cancer Center. So we tested our TNF in a test tube. It worked beautifully as anti-cancer agents. And then we tested in animals. It worked beautifully. But turned out that humans are neither test tube nor animals. Humans are human. So until we test our TNF in cancer patients, we don't know whether it will work or not. So that is where I got recruited by University of Texas MD Anderson Cancer Center to test our TNF in the cancer patients, and they did. And virtually all the patients responded, but they got more sick than ever before. So what was necrotizing tumors in the test tube or in animals, it was promoting tumors in the animal. Just imagine. So it was making patients worse. So that is the time they started making inhibitors of TNF. So here is the pharmaceutical companies. They came up with the small molecules as inhibitors of TNF. And apparently these are some of the diseases that have been linked to TNF, and TNF inhibitors are used for it. And now that has created a market over $40 billion a year. Billion. And that is for the course of last 30 years. And no drug known to man, I challenge you, has a bigger market than that. And just so happened, and as I was a kid in Punjab, where we came as a refugee from Pakistan in 1947, my family did, and I was born in India, in Punjab, and my mother, that anytime you get hurt, anytime you have any kind of issue, the first thing that she will apply is haldi, turmeric. And so I had this very strong passion towards turmeric, that it works for all kind of diseases, all kind of ailments, here, there, and whatnot. So I did the same thing with the TNF, that will turmeric block TNF? And the answer is yes. So we were the first one to show that yes, indeed, that turmeric will block TNF, and this $40 billion market that which is antibody against TNF that can be counteracted by turmeric. Question is, what is there in turmeric that can block TNF? So that is where we were the first one to identify a compound from turmeric called curcumin that will block TNF. So I'm going to show you, and so these are some of the drugs that have been approved as TNF inhibitor by the FDA. And so FDA is a very, 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 very crazy organization, to tell you the truth. And the reason for that being that FDA approved all these things as TNF blockers for various diseases. So what we did, we formed a company called Curry Pharmaceutical. Under that, we went with the curcumin, which is the yellow color in turmeric, to the FDA for approval. And FDA declined. And why they declined? They said, look, people have been eating for thousands of years. Why do you need my approval? Just go home and eat as much as you want. Okay, so they completely declined. So as of today, it is used by a lot of different companies. I already mentioned that the market for TNF blocker is $40 billion. And this is data from 2017. And these are some of the companies that are making TNF blocker, which is all antibodies. And so all the blockers that are approved by the FDA, they have black label warning, which means they have a lot of side effects. And these are all side effects that I mentioned. And that means they are not good for you. And yet people are willing to pay $40 billion. And gradually, and very gradually, they are beginning to realize that turmeric can do exactly that. Now there are a lot of shops in America. They are selling turmeric and curcumin in all different shapes and forms and whatnot. So much so Johnson & Johnson made even a Band-Aid. As I mentioned that when we were kids, we get hurt. The first thing mother will apply was turmeric. So Johnson & Johnson even made a Band-Aid that if you get hurt, you can make a turmeric Band-Aid. So I think I don't want to kind of confuse all of you. So we have written a lot of reviews, a lot of papers that you already heard from Madan. And here is one of the paper, if anybody is interested, can look it up, that both upside and downside of TNF blockers. And when I say downside, that I already mentioned, there's a black label warning given by the FDA for all the TNF blocker, but nobody yet, to my knowledge, has given a black label warning to turmeric. So turmeric is very, very safe, and it works remarkably well. So, and then if you look at the global cancer incidence, no country has more cancer than America. And no country has spent more money to find a cure to cancer than America. So here you can see in red that shown over here is the highest incidence of cancer. And then India has one of the lowest incidence of cancer in the world, mind you. However, as India is becoming Americanized, cancer is going up and up and up, even in India, because they are trying to adapt the American lifestyle. And the future is looking even more oblique, both for India and America, because as I mentioned, mother is the one that brings us in this world, and mother nature is there to take care of it. More you move away from the mother nature, more you are going to pay heavy price for cancer and all the other chronic diseases. So, and that is what is happening. So here you can see that the projection for global cancer incidence and cancer death. And you can see that what is shown in red is cancer death, and what is shown here in yellow is the new cases of cancer. And you can see that 2002, it's not working. So you can see that 2002 versus 2020 and versus 2030, that cancer incidence is going up and up and up. Okay, and the cancer death here shown in red is going to go up and up and up. So as I said, more you run away from the mother nature, more it is not in your favor as indicated by this slide. And here is India alone. And this is the most recent data. And you can see the cases in 1990 versus 2016. And you can see state by state the burden of cancer and the variation across the states of India. Again, it is going up and up. And I think not very many people know this, but I want to share with you that one of the first drug ever discovered for cancer and approved by the FDA was Indian people. And here I'm showing you 3 different people who you may or may not have heard of, that who discovered the first anti-cancer drug known to man. And you can see Subbarao, you can see Sahgal, and you can see Mansukh Wani. And I showed there, I'm showing you a picture, and I'm showing the drug that they are the one who discovered. And they are used even as of today. And all 3 of them were in America. Mansukh Wani spent most of his career in America, and Subbarao was in Chennai for a while, and Sahgal was in New Jersey. So kind of gives you an idea that this is where the first anti-cancer drugs came along. And as of today, these are all the drugs that have been approved by the FDA between 1949 and 2014. And look at the number of drugs. And curcumin, you will be amazed that how much in parallel it can go with these drugs. So, okay, so now you got all these drugs and you got billions of dollars. Question is it helping? And here is the latest paper that came in 2017 in Cell, that of the 91 new therapies approved for solid tumors between 2002 and 2016, the median overall survival benefit was little more than 2 months. Only 2 months after spending billions of dollars. Yet the annual price tag per patient now is regularly exceeds $100,000. It's not I'm saying it. This is a paper coming in Cell. Only 2 months increase in lifespan. It's making a fool of somebody. Okay. So here is a food for thought, and that's what we are here for. And so food for thought, how complicated is the human disease? We need to understand that. And apparently it is very, 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 very complicated. And I won't have the time to go through it. And there are a lot of other facts about the DNA and so forth. I cannot see from here very well. But I think, as I said, all this information is available to you. And please make use of it. So our working hypothesis has been that dysregulated chronic inflammation. Inflammation is Agni Devta, fire god. If fire is under control, everything is good. And if fire is out of control, everything is bad. So our whole immune system is full of fire. And as long as the immune system is under control, no problem. But same immune system, when it gets out of control, it causes havoc. So, and that's what dysregulated chronic inflammation. And caused by lifestyle factors, mediate chronic diseases, including cancer. So there you have the solution. You control inflammation, you control the disease. That's it. And here is the inflammation. It is a fire. Whole issue of immunity devoted to it. Okay. So here is the secret killer. The whole issue of Time magazine, okay, is about inflammation, fire, the secret killer. And the fires within, you have seen this slide even before. Okay. These are fire burning even while we are sitting over here. Different people are thinking of different things. There are different things bothering to different people. So different kind of fires burning in all of us. And question is how to keep it out of control. And some are able to, while others are not. They get out of hand. So these are all fires. And so here is the controlled fire shown on the top. As I said, fire is not bad. And so here is in the kitchen, in the living room, in the bedroom, you have a fire on, everything is good. And here at the second part is uncontrolled fire. Same fire, same inflammation out of control. You can see it causes havoc. So as long as you keep your inflammation under control, you are fine. So question is where the inflammation is coming from to begin with. Here you can see cigarette smoking. 30% of all cancer is due to cigarette smoking. Here is a radiation. At least this is the problem that does not exist in India, that where all these women, men sitting under the radiation, trying to burn their skin and ending up in skin cancer. Having known that all this radiation will cause skin cancer, still people in America or Western world, they do it. And then here is a diet, here is obesity. These are all lifestyle carcinogens causing inflammation. So here are further various sources of inflammation that I have outlined: environmental pollution, food factor, bacteria, viruses, and stress. People are more under stress now than ever before. It means inflammation. Ultraviolet radiation, alcoholic beverages, these are all inflammation. So here, as you can see, that I listed that cancer is a preventable disease that requires change in lifestyle. And 30% of all cancer due to tobacco, 35% diet. As they say, you are what you eat. So if your diet is not right, it will lead to cancer. 35% of cancer due to diet. And 14 to 20% is obesity, 18% is infection, 7% environmental pollution, and only 5% and max 10% is due to the bad gene that you inherit. So you can very well imagine that most people who get cancer, where do they get the cancer from? And we need to know that. And mind you, that I hate to say that even a lot of medical doctors that I have ran into, they do not know about it. And they don't want their patients to know about it either. And I have several medical doctors come to me that even if they know it, they will use it for themselves, but they will never use it for the patient because they cannot make any money out of it. Okay. So here you can see nearly 50% of cancer deaths worldwide. And this paper came very recently, 2022, August 31st, 2022. Nearly 50% of cancer deaths worldwide are caused by preventable risk factors like smoking, alcohol, and obesity. So the data is all there. This is global cancer death as of 2022. So there is a word called ETs. And it is this guy, his name is Rudolf Virchow. In 1850, he was the first one to define inflammation and its link to cancer. And he is from Würzburg, Germany. Here we have somebody from Germany, from Berlin. Oh, I don't see him here in the audience. And so apparently it was in Germany that the first definition of inflammation, and it's linked with the atherosclerosis, is linked with the rheumatoid arthritis, multiple sclerosis, cancer, asthma, Alzheimer, you name it. So it was this guy was the first one to do that. So he was also the one to define inflammation as a pain, heat, redness, swelling, and loss of function. And that was the definition of inflammation. And he gave the word inflammation introduced as ETs. If somebody says, I have arthritis, which means inflammation of the joints. If somebody says, I have a bronchitis, inflammation of the bronchus, or sinusitis. So wherever this ETs appears, it means inflammation. So as of today, there are 200 different kinds of ETs. Okay, here are all the ETs for you. These are all inflammation. Okay. So Science journal that all of you know is a very, very prestigious, devoted their whole issue to inflammation. And this is copy from 2013. And now, as of 2021, again, whole issue of Science is devoted to inflammation. So if anybody is interested, can look it up. Okay, that how critical, how important is inflammation. So here are all the different cancers that I have indicated here, that which are caused by inflammation. I have indicated here that where the inflammation is coming from. And then I have indicated that, say, bronchitis leading to lung cancer, hepatitis leading to liver cancer, cervicitis leading to cervical cancer, colitis leading to colon cancer. So all this ETs leads to cancer. Okay, so wherever this word ETs means, it end up in cancer. So therefore, our working hypothesis has been that NF-kappa B activation is a major mediator of inflammation in most chronic diseases, including cancer. And if you were to inhibit NF-kappa B, you can prevent and even delay the onset of chronic disease. It is that simple. Okay. So these are all the genes that are regulated by NF-kappa B. So by just regulating one transcription factor, you can regulate all these various genes. So just NF-kappa B. And I'm going to show you that turmeric can do just that. Okay. So here is NF-kappa B. So TNF, everything bad that TNF does is through NF-kappa B. And you can see that I indicated NF-kappa B further lead to STAT3, and it is involved in survival of the tumor, proliferation of the tumor, angiogenesis, invasion, metastasis, bone loss. Every aspect of cancer is regulated by NF-kappa. So these are just all the different where NF-kappa B is coming from. The tobacco-linked cancer, carcinogen-induced cancer, virus-induced cancer, UV-induced cancer. They are all mediated through NF-kappa B. So NF-kappa B addiction and its role in cancer. One size does not fit all. So this is the role of NF-kappa B, and we did a whole review that was published in Oncogene. And these are all the different genes that are regulated, the crosstalk between NF-kappa B and other transcription factors. And I'm running short of time, so I'm going to kind of quickly go through some of the other stuff. And here just to show you that virtually every stage of cancer, from normal to tumor, survival of the tumor, proliferation, invasion, angiogenesis, metastasis, they are all regulated by NF-kappa B. And most cancers start at the age of 20, mind you, and they're manifested at the age of 40. Okay, between 40 and 50. And so here I have indicated that 10 to 20 years is its incubation phase, and another 20 years for its progression phase. So these are NF-kappa B is involved not only in the immune system, and here I have indicated all the various diseases that have been linked to NF-kappa B. And as I mentioned, NF-kappa B is a transcription factor, causes inflammation, and here are all these various diseases linked to inflammation. And the reason I'm going through all that, I'm going to show you a whole bunch of natural products that can block just that. It is that simple. So here is with Ajay Kunnumakara and all the others that we have done another review, inflammation, NF-kappa B, and chronic diseases. How are they linked? And pretty much spelled out here. And so, for example, cigarette smoke. Cigarette smokes activates NF-kappa B and uses COX-2. So that's where NF-kappa B is coming from. And here we have looked at lung cancer patients, and as the cancer gets more and more advanced, there is a more and more NF-kappa B. And here is obesity. And as I mentioned, that 14% of the cancer in male and 20% of the female have been linked to obesity. And here are all the cancers that have been linked to obesity. So NF-kappa B is the enemy within us. And we published a paper in Cancer Cell. NF-kappa B can be a friend if it remains in the immune system. But moment it gets out of the immune system, it becomes a foe. And that's what we have this review. NF-kappa in cancer, a matter of life and death. Okay, so one has to remember. So here we have a stress, NF-kappa B, inflammation, and leading to cancer. So this has been our working hypothesis over the years. And inflammation, a double-edged sword for cancer and other age-related diseases. This is another review that we did together with Subhash Gupta and Ajay Kunnamakara and Sadhana Agarwal. I think all three of them are sitting in the audience right now. And so they are the one drivers of this review. So this is again the same thing. And here is a neutralizing tumor promoting inflammation, a magic bullet. And this is again my colleague Lisa Cousin and from UCSF did a whole review for Science that neutralizing tumor promoting chronic inflammation, a magic bullet. So question is how to control inflammation. There are steroids that can control inflammation. There are NSAID, non-steroid anti-inflammatory drugs. There's Celebrex, there's a metformin, there's statins. And these are all drugs that have been approved by the FDA and they can all control inflammation. However, this is a conference on nutraceutical, and then the whole bunch of natural products that can control inflammation. And that's what we're going to talk about. And these are the natural products. These are, spices. And as I mentioned, India is a land of spices. These are Ayurvedic medicines. These are fruits and vegetables, traditional Chinese medicine, and so forth. All these can control inflammation. And here are the active compounds that I have outlined for you, that what is there in all those spices and Ayurveda and so forth that can control inflammation, and it is already documented. And these are the compounds. So this is nothing different because Hippocrates proclaimed 25 centuries ago, let food be thy medicine and medicine be thy food. It is that simple. And so simply what we are trying to do is scientifically prove it that whatever Hippocrates said 25 centuries ago is true. Okay. And here again, we have another review. There's a whole book that we edited, Inflammation, Lifestyle, and Chronic Diseases, a silent link. And here is a farmer's market, and you can see the fruits, spices, vegetable, cereals, that active compounds have been identified that can control inflammation. Here is even a bigger farmer's market, and it's keep growing. Here is a more bigger farmer's market, and these are all fruits and vegetables that can control inflammation, and the active compounds have been identified. And here is a more. And here, for example, resveratrol is one of the agents we have identified that can block NF-kappa B, can block inflammation. So question is, what is resveratrol? And here I showed you the structure and where it is coming from. I showed you all the different kind of plants and that actually produce resveratrol. And grapes, for example, that be that red grapes or white grapes, they produce resveratrol. And grapes lead to wine. If it is a red wine that people which they love, and they are acquiring resveratrol. So I indicated over here. So resveratrol addiction, to die or not to die. And when I say resveratrol addiction, because a lot of people, they have a wine. So they are going for a wine for resveratrol. So they are addicted to the wine. Why? Because they think this is how they get resveratrol. Question is it going to do them any good, or is it going to do them any bad? And that is where the symbol comes in, to die or not to die. Resveratrol, a multi-targeted agent for age-associated chronic diseases. It blocks NF-kappa B and blocks a lot of other things. And chemosensitization of tumors by resveratrol. Again, you can look it up. And Subhash Gupta, sitting right here, is the one who did this review. So here is more that Shakibai, you already heard Shakibai also talk about it. And here is indole 3-carbinol. another compound that coming from the mother nature. And here I indicated all the different fruits and vegetables where it comes from. Again, blocks NF-kappa B and works remarkably well for all the various chronic diseases. And again, all this should look very familiar to you. So here is a tocopherol. So everybody heard of tocopherol, but here is a tocopherol, the vitamin E of the 21st century, and its potential against cancer and various other chronic diseases. And so this is a tocopherol, which is vitamin E. And so a lot of people have heard of vitamin E, but very few people have heard of tocopherol, which is the brother or sister of tocopherol. And that is shown here in the red in terms of these differences in structure and so forth. And so this is where tocopherol comes from. And as you can see, and in Malaysia is very big on all this palm oil, so palm, rice, barley, oat, wheat, and so they all are the sources of tocopherol. So here is a more further sources, and I can go on and on. And so that is all sources of tocopherol. So here is that we published an article that tocopherol, but not tocopherol, inhibits NF-kappa. And it is all spelled out as it's the rest. So in other words, even though they have a similar structure, but they are not the same, they behave very differently. So role of tocopherol in the treatment of human pancreatic cancer. And we looked at it and other people looked at it. And here you can see gamma tocopherol inhibit pancreatic tumors and sensitize them to gemcitabine, which is the standard treatment for modulating inflammatory microenvironment. So it is that simple. So you can use chemo all right, but you can make chemo more effective if it is combined with tocopherol. And that's what this is about. And this is again, Kunnu Makara is the driver for that. And Ayurveda, and as all of you know, that India is the land of Ayurveda. And Ayurveda means science of long life. And the origin of Ayurveda dates back to as much as 5000 BCE. And it originated as an oral tradition. And Ayurveda consists of Chark Samhita, Sushrita Samhita, and Bela Samhita, written all around 6th century BC. And that is again very strong part of our culture. And we did a whole review from traditional Ayurvedic medicine to modern medicine. Okay, identification of therapeutic targets for suppression of inflammation and cancer. And you can see that some of these people here, and you heard Gautam Sethi, he just gave a talk today, and he's a part of this. So here is identification of novel anti-inflammatory agents from Ayurvedic medicine again for chronic diseases, reverse pharmacology. And so just to give you a kind of preview, this is a Google. So we did a whole review, Googling the Google. Okay. And Google is nothing but Withania somnifera. Here I showed what it looks like, identify the active compound, and it blocks the NF-kappa. Googling the Google for prevention of chronic disease. And again, Ajay Kunnu Makara is the driver for this particular review. So here is another one that where we showed that it completely blocks NF-kappa. So, and this was published with Ichikawa way back then. Here is a boswellia, and it's also called frankincense or salai guggul. And here is an active compound that in salai guggul, and it again blocks NF-kappa. Here is a ashwagandha, Withania somnifera, withanolites. Ashwa means horse, gandha means smell. It smells like a horse. That's why I call ashwagandha. Okay, and Withania somnifera, and the active compound is withanolite, and again it blocks NF-kappa. Here is a long pepper. Everybody heard of small pepper? And here is a long pepper called Piper longum. It selectively kills the cancer guy, but not the normal guy. And the mechanism by which it works. And this whole article came in Nature about Piper longum. And we published that this is working through NF-kappa B. Okay. And here is a neem. Neem is Azadi Raktika indica. Azadi means freedom. Drakt means tree. The tree that gives you freedom from all diseases. Azadi Raktika. Indica means from India. So this is a Sarav Rog Nivarani is the name in Sanskrit for neem. And again, the active compound is nimbolite. And this again tree that not all of you may know, that is very, very, very native to India only. And it works remarkably well. And here we published again in JBC that how this neem compound nimbolite works against cancer. And again, Subhash Gupta is the driver for this particular article. And here is again another article published by Subhash Gupta on neem compound. So from exotic spice to modern drugs. And this is another Dr. Singh published in Cell in 2007, that the global demand for more affordable therapeutic and concern about side effects of commonly used drugs are refocusing interest on Eastern traditional medicine, particularly those from India and China. And so here we did a review again, Ajay Kunnumakara, chronic diseases, inflammation and spices. How are they linked? And these are all the various spices what we are talking about. I don't have to tell you, you guys know very well. And here is a Vasco da Gama looking for spices. And the route that he took is called Spice Route. And here is the whole avenue that indicated, and in Goa way back in 1498 that he came here looking for spices. And these are the spices that what he was looking for. And we have a whole book, if anybody is interested, called Healing Spices. Over 100,000 copies are already sold. And these are already translated into different languages. So which deals with spice by spice by spice. So, and another book that we have here that you can see, molecular targets and therapeutic uses of spices. And again, if anybody is interested, Ajay is an integral part of this. And here is add spice to your life. And that's what we are talking about. Curry in a hurry. You know, they opened up a restaurant in New York. Okay. And spice it up. Spice queen. These are the terminology that is used in America right now. And spice goddess. You know, so I'm trying to, you know, show you that how they use spices over there. So much so most of the American women, they are named after spices. How? Here you can see anise, ginger, rosemary, mace, pepper, basil, tulsi, sage, jasmine, angelica, curry, chili, tamarind. These are all American women. They are all named after spices. They may not eat the, you know, spices, but they named all the women after spices. Okay. So here is a, you know, another guy, Manoj Pandey, and even Subhash Gupta is a very much part of it. Dietary nutraceutical as a backbone for bone health. Okay, to keep the bone strong, healing with spices. So these are all the various spices, and that's what we are talking about. And here are more spices. More spices I don't have to tell you, you know very well. And curcumin is one of the spices we publish in annual reviews of nutrition, targeting inflammation-induced obesity and metabolic diseases by curcumin and other nutraceuticals. And molecular targets of nutraceutical derived from dietary spices. And this is another review that has been done. And here is a curcumin that it is coming from, where it is coming from, capsaicin and diacetyl and anethol, eugenol, ursolic acid. These are all coming from different spices, and they all block NF-kappa. And here is a red chili, and it blocks NF-kappa B, and shown in all this publication. Here is a ginger, blocks NF-kappa B, and shown in here. Here is a ginger, it prevents bone loss. So here is a lot of experiment that have been done. So if anybody has a bone issue, and take more ginger. Here is a black cumin, and again blocks NF-kappa B. Here is a fenugreek, again blocks NF-kappa B. Here is a fennel, again blocks NF-kappa B. Here is a black pepper, again blocks NF-kappa B. Here is a cardamom, blocks NF-kappa B. Here is a cardamom again, rank ligand, which is an antibody against rank ligand, have been approved by the FDA with market several $1 billion. And cardamom just can do that. So nobody going to make any money out of cardamom as compared to these antibodies against this. And here is a curcumin, getting back to our roots. And that's what we are here. And this is the structure of curcumin. And this is just to show you that where it is coming from. And this is, we have a whole issue, trends in pharmacological sciences, that they put us on the cover on this magazine called Pharmacology of Curcumin. I'm sure there are some people here from South India, and in South India, they use a turmeric even to color their feet. And that is shown right over here. And so they put that one on the cover of this particular magazine. Okay. And so they wrote our whole review on this pharmacological basis for the role of curcumin in chronic diseases and age-old spice with modern targets. Okay. So, and apparently there are 100 different types of turmeric, over 100 different types. And here I happen to be hanging around in Chitrakoot, and Chitrakoot, they were selling black turmeric. And in Gujarat, there is a white turmeric. And Karkuma amba means it tastes like a mango. Okay. And there is a black turmeric, and they use it to induce abortion and all kind of things. And I was in, Turkey, again, they had black turmeric. So there are 100 different kind of turmeric, but we know very little about all the others. So it was this particular guy, and the first paper ever to be published on curcumin was in Nature. So as an antibacterial agent. And here is that paper, and this is a guy by Vogel, and apparently it turned out to be, as of today, that turmeric curcumin is antibacterial, antiviral, antifungal, anti-cancer, anti-obesity. The list goes on and on. And even anti-coronavirus with Ajay Kunnumakara, we did the whole review on that one, that it completely knocks down a coronavirus. Okay. And so as I said, list goes on and on. And here is just to show you that the amount of curcumin that is present in turmeric, if it is a 25 gram of turmeric, half a gram will be a curcumin in it. And as I already mentioned, Johnson & Johnson has made a Band-Aid. And here I'm showing the Band-Aid. And there is a Vicco Vajradanti, which already all of you know is a curcumin paste. And there, before pH meter came along, turmeric was used as a pH indicator. How? That if it is put under acidic condition, turmeric will turn black. If it is put under alkaline condition, it will turn red. So you can tell the pH of a solution just by using turmeric. So here I showed all of that. So antibacterial action, as I mentioned, this is published in Nature in 1949. So here is just to show you that this is our first own paper on curcumin as anti-inflammatory agent, and it completely blocks NF-kappa B. And it just, all the data that is published, and any of you can look it up. And here, so far, nobody, I challenge you, nobody has found a cancer that curcumin cannot kill. And here are all the cancers that I have indicated, at least in the test tube, at least in the animals, but now at least in humans. So together with Ajay Kunnumakara, it turned out that over 500 clinical trials now have been done with the curcumin alone. And normally one clinical trial is enough to get an FDA approval. And 500 clinical trials tell you everything that what curcumin does in humans. And here are all the different inflammatory targets of curcumin. And here is all the different things curcumin binds to. Mind you, this is a physical binding of curcumin to all these different things in our own body. It is mind-boggling. Okay. And so curcumin is orally bioavailable blocker of TNF. The blocker of TNF that has been approved by the FDA, which are antibody, have to be injected. And this one is orally bioavailable. What more you can ask? So here is a woman, Usha Rani. I don't know how many of you know. She's from Nizam Institute in Hyderabad. And she is the one who showed that if you give 150 milligram curcumin twice a day, and here she give it to people, 21 people placebo and 23 people here with curcumin, you can see serum levels of TNF are down. So now we are not talking about rat or mice. We are talking about human, and 150 milligram and no modification. So a lot of people have modified curcumin. Why? Because they're looking for free curcumin. So when they give it to human, they don't see anything free, because curcumin has so many levers in our body that it is not freely available. So they think it disappeared. But question is, what is the end result? End result is shown right here. It does what it is supposed to, blocks TNF and a lot of other things. But it's not freely available because there are so many levers of curcumin in our body to which it binds to and then delivered wherever it is needed. So these are all the clinical trials at 120 listed over here, and even it became even longer. Now this is over 300. As I said, normally one clinical trial is enough to get FDA approved. Over 300. Okay. So here is a biliary diseases published by Albert Oppenheimer in Lancet in 1937. Just imagine. Okay. And phase 2 clinical trial curcumin with advanced pancreatic cancer. This is what my colleague Rizal Kurzra, we published together with her, that it works remarkably well for pancreatic cancer. And as all of you know, pancreatic cancer is very, very lethal cancer. Okay. And even a guy like Steve Jobs died of pancreatic cancer. Okay. And so here is a therapeutic role of curcumin, lesson to be learned from clinical trial. Again, Subhash Gupta is the driver. And here are all the different diseases where curcumin has been tested. And you can see the list is very, very, very long. And the curcumin bioavailability, how big is the problem? And here we have published a lot of papers on curcumin bioavailability, and I'm not going to go through, but the bottom line is there's no problem at all. And here you can see in the blood, thyroid, heart, lung, liver, spleen, the kidney, muscle, and I indicated it in matter of minutes, and it is everywhere in our body. No problem whatsoever. And they have made even a Band-Aid. And here is a topical application. I happen to be giving a talk in Bulgaria, and apparently, one of the guys from the audience who was a medical doctor came to me, and apparently he said he's using the curcumin for cutaneous T-cell lymphoma. He said he's topically applying on this patient, and he gave me actually pictures, and I'm sharing these pictures with you. Topical application, okay, and works remarkably. So this is even psoriasis. There's a woman at UCLA, and so she's using curcumin for psoriasis. And here you can see that this is the picture that she gave me, that here you can see topical application. She even changed the name of curcumin to Soria Gold. So she formed a company, and she's selling curcumin under the name of Soria Gold because it blocks psoriasis. Okay. And so her name is Madeline Hang, and I indicated over here. So it works remarkably. And here is a uveitis, and which is again inflammation in the eye, and it works remarkably well. And so, and it is very synergistic with the resveratrol, green tea, quercetin, piperine, genistein, you name it, and you can use it. So I think that the bottom line is that India is a land of spices. It is a land of Ayurveda. Therefore, if you look at the incidence of cancer in India versus United States, and I indicated over here that spice consumption here is shown in red, and here is incidence of cancer in male shown in red, and incidence of cancer in females shown here in green. And this is India, and here you look at versus US, that the amount of spices consumed is very, very low, and then incidence of cancer in male versus female is very, very high. It is not in your favor. So more spices you consume, lesser is the cancer. And that is the bottom line. And this is a data coming from NIH, where they are looking at the epidemiological evidence. And here, then they have gone cancer by cancer in US versus India. The number of cases versus number of deaths. And you can see it for yourself. So it just so happened that I happened to be giving a talk in Turkey, Istanbul. And there is a grand bazaar in Istanbul. And right outside the grand bazaar, I see my name, Bharat, written over here. And the guy has all the spices over there. And my wife is taking my picture. Here I'm standing here. And the guy who is the owner of the shop is objecting that why you are taking a picture of my shop. And my wife said, the reason I'm taking his picture because his name is Bharat too. And he said, oh, impossible. How could he be Bharat? Bharat is spicy. So to him, Bharat means spices. And that's why he named his shop as Bharat. So apparently in Turkish language, Bharat means spices. Okay. That's something which is kind of, is it a coincidence or is it luck? So haldi is healthy. And we've done another review. And as I mentioned, in New York, they opened up a restaurant. In Curry Hill, a kid on the block called, the name of the restaurant is Haldi. And in Palo Alto, Stanford, they opened up a restaurant called Turmeric, the golden spice. Okay. And this is just to show you turmeric cookie, turmeric latte, all kind of things now they are putting turmeric. These are all the different products that are based on turmeric. So it is getting very, very, very popular. And so even I mentioned that we did a review with, this is Ajay Kunnumakara, COVID, cytokine inflammation and spices. How are they related? And it is published in 2021. And this is a Nature review. And I just don't want to kind of go through it. And with all that. So now because of all these spices and because of our contribution to Ayurveda, if you look at 200,000 scientists in the world, we are rated as number 84 on this one. And as you heard earlier, that all because of the citation index. And with that, I just want to tell you that anything and everything that I have said till now, I'm not the doer. The doers are all these people described. There are several of them here in the audience, and they are the one who carried out all the work. And I just oversee it. And so I'm very thankful to all of them for doing all this. So with that, I stop right here and thank you.
Thank you, sir, for a very, very elaborate talk and enlightening us on the our heritage and the future use of the spices, both in therapy and prevention.
Thank you.
Thank you very much, Professor Agarwal, for a very informative and invigorating lecture. I would now request Professor Chaturvedi to felicitate Professor Agarwal with a shawl and a memento. I would now request Professor Kishore Singh to present a shawl and a bouquet to Mrs. Uma Agarwal. I would also request Professor Dr. Kishore Singh to present a memento to our chairperson, Professor M.M. Chaturvedi. I would request Professor Alok Bharti sir to present a flower bouquet to Dr. Kishore Singh. Thank you very much, ladies and gentlemen. We would right now begin with the valedictory session. And for that, I hand over the stage to Apurva.
A very good morning to all the attendees, professors, scholars, and students. I, Apurva Chaudhary, warmly welcome you all for today's valedictory session of Incd 2022. Hope you all have enjoyed our 3-day conference. Without delaying the event, I request Professor Bharat B. Agarwal to come upon the dais and present a bouquet to our honorable chief guest, Professor Kishore Singh. Bouquet. It's all right. I request other dignitaries to come upon the dais. Professor Alok Chandra Bharti, Organizing Secretary. Professor Shriji P, Co-Convener. Professor Ajay Kumar Kundamukara, Co-Convener. Professor Raja Gopal Raman, Co-Convener. No. Professor Subhash C. Gupta, Co-Convener, and Professor Anju Srivastava, Organizing Secretary of Incd 2022. I request Professor Anju Srivastava to read the report of the conference.
Thank you, Apurva. And it's my pleasure and duty to actually read the report. I'll just give a brief account what all happened, how the conference was actually conceived, from where we started and where we have ended. And I think all of you have enjoyed. This day was good in spite of continuous rain, but in some way it was good. It forced all of you to stay in the room, and it's good to see, at least at this point in time, there are many people sitting in the room. So I'll just read out the report which has been prepared, and this is this 5th International Conference on Nutraceutical and Chronic Diseases on pharmaceuticals and nutraceuticals for cancer and other chronic diseases from 7th to 9th October was organized by Department of Zoology on the occasion of centenary year of Delhi University and 75th year of Department of Zoology. And the conference was actually organized under the umbrella of Society of Nutraceutical and Chronic Diseases, Society of Translational Cancer Research, and Evolvera Society. It was designed to explore the scientific basis of experimental approach for the use of pharmaceuticals and nutraceuticals for cancer and other chronic diseases. It was initiated in 2016 with first meeting at Cochin, and as Sir had already mentioned, there were consecutive several meetings in different places of India. And after COVID-19, it was our opportunity at Delhi University to actually organize this conference, and it was our pleasant duty. We felt very, very privileged. With the core organizing committee consisting of 54 members, both from both national and international, 10 different thematic areas were finalized, such as, to name a few, therapeutic potentials of nutraceuticals in stress and cancer, in neurological and neuromuscular disorders, molecular targets and drug delivery, cancer genomics, metabolic disorders and personalized medicine, emerging anti-cancer phytonutraceuticals for chronic diseases and chronic infectious diseases and emerging non-infectious diseases, are used therapies for chronic diseases and inflammatory disorders. The conference preparation actually started with the call for invited lectures from distinguished speakers all across the globe. And it was a difficult time, and it was very heartening to see the enthusiasm of participation. There were many people actually came forward to this conference. There were more than 100 actually speakers, both from nation and number of other countries. Entire India was actually represented, and most of you must have actually got the opportunity to interact with the participant. There were 124 participants comprising of 34 oral presentation from post-PhD scholars or postdoctoral fellows, research scholars, postgraduate students, and graduate students. There were 63 presentations from the students. Finally, the designing of the scientific program to accommodate all the participants was a very, very tedious job because there were a number of participants, and we never wanted actually parallel sessions to be organized, but because of the overwhelming response, we had to actually make parallel sessions. And in some cases, some of the talks you might have missed, but all those talks are actually on YouTube. It was live streamed. So most of you, those who have missed it, can actually see it again from the YouTube. The conference actually had 6 parallel sessions, 13 keynotes, 51 invited lectures, and a highly academic enriching panel discussion. The conference INCD 2022 was also organized with an aim to promote the use of nutraceuticals. For this purpose, we invited the companies, nutraceutical companies, to showcase their products to boost the usage of these efficacious molecules. We are delighted to announce that this nutraceutical exhibition saw enthusiastic participation by 8 different nutraceutical companies, namely Merck Scientific Private Limited, Thermo Fisher Scientific, Becton and Dickinson, and Sami Biorad, Incide Private Limited, Nikon, Fujifilm, Vaco Pure Chemical Corporation, Eppendorf, India. Collectively, with online delegates, this conference saw humongous participation of more than 200 nationally and international participants. The first day of the conference began with welcome and opening remark followed by plenary session by DILAB on therapeutic potentials of nutraceutical, the inaugural session chaired by Professor Reena Chakravarti and Professor Kalpana Pai with Professor Yogesh Singh, Honorable Vice Chancellor of University of Delhi, and Professor Bharat Bhushan Patwardhan, the Honorable Chief Guest of the INCD 2022, turned out to be a memorable one. Apart from this, there was a heritage walk to Shahid Bhagat Singh Smarak emerged as a highlight of the conference. The first day ended with 2 interactive parallel sessions in 3 different halls covering nutraceuticals and neurological and neuromuscular disorders, molecular targets, drug delivery, cancer genomics, metabolic disorders, personalized medicine, emerging anti-cancer phytonutraceuticals, and oral presentation by research scholars. The second day started with parallel sessions, plenary sessions, and was graced with interactive and informative public lecture by Professor Acharan S. Narula. The scientific program closed with an interactive panel discussion moderated by Professor Subhash C. Gupta. Finally, the cultural event organized by Evolvera Society at the Department of Zoology to commemorate completion of 75 years of Department of Zoology emerged as a lively highlight of such a busy scientific day. Finally, the third day opened with parallel sessions and young faculty oral presentation. The impressive poster presentation by research scholars was the hotspot where educational information was concentrated, was highlighted, was actually for highlighting the research on the bench side. The hard work of the participating students was encouraged shortly, will be encouraged shortly by number of presentation, award presentation, poster award presentation. And in addition, there are 10 travel awards which are going to be announced in support of young investigators. These 3 days turned out to be a magnificent academic journey where we met such eminent speakers from all around the world. This is not the end of the scientific journey, rather it can turn to be a new beginning of the scientific avenues, especially in the nutraceutical science. So I think this actually completes the story which we have actually, or the journey of last 3 days. And I hope all of you have enjoyed and gotten enriched. So thank you very much.
Thank you very much, ma'am. I request our Honorable Chief Guest, Professor Kishore Singh, to address our today's lovely gathering.
Namaskar. At the outset, I want to thank the Bharti ji for calling me. It's an honor and privilege to address the gathering. As we are progressing in the modern times, our longevity is increasing, and so is the population. So cancer is going to be center stage in coming years, reflected in increase in the incidence. Not only incidence will increase, also the pattern of cancer will going to change. The known tobacco-induced cancer is going to reduce as we are quitting tobacco, but the other cancers like pancreas and different lifestyle cancer will going to increase. So that will bring the nutraceutical into the center stage to control these lifestyle-induced cancer. Breast is already #1 in India. Cervical cancer is going down with each passing year. So more and more we are going to face this type of problem, and this makes the importance of this conference much more to bring into focus the research on nutraceutical and diet and lifestyle in the management and prevention of the cancer. With these words, I thank you again.
Thank you very much, sir. I request Professor Anju Srivastava to announce the results and Professor Kishore Singh to present the awards to the prize winners.
Okay. कौन सा है postdoctoral session? So the first category of award is for postdoctoral session or for postdoctoral students. The first award goes to Dr. Tejveer Singh from Jawaharlal Nehru University, New Delhi. Congratulations, Tejveer. The second prize in the same category goes to Dr. Akanksha Singh from CSIR Institute of Genomics and Integrative Biology. CSIR, Institute of Genomics and Integrative Biology, New Delhi. IGIB, okay. The second category is doctoral session, the PhD students. The first prize goes to Alicia Walson, C, from CSIR, National Institute for Interdisciplinary Science and Technology, Thiruvananthapuram, Kerala. The next award goes to Ayushi Garg from Punjab University, Chandigarh, Punjab. Okay. The third prize goes to Regina Tennyson from Rajiv Gandhi Center for Biotechnology, Thiruvananthapuram, Kerala. The fourth prize in the same category goes to Kulbhushan Thakur from University of Delhi, Department of Zoology. And the fifth in the line goes to Saima Saida from Department of Zoology, University of Delhi. For poster presentation group, on day one, 2 students were selected for the award. Ashwati M. Ashwati, yes. CSIR NIST Thiruvananthapuram, Kerala. And the second, NIST from NIST. Vidushi Tyagi from CSIR CMAP. Day 2, Anna Sandram, Daulatram College. Anna? She's not here? Okay. Anyone who wants to take her on her behalf? Okay. Okay. Next one is and for day 2, another one, the most, I think all of you are going to clap for her very loudly. That is Nikita Agarwal, Department of Zoology. And I think all of you will agree, she was our Annapurna for these 3 days. And on the 3rd day for today, it is C.K. Kirthana, RGCB Tiruvananthapuram. And the next one is Vaidehi P. Thakur from GCRI Ahmedabad, Gujarat. Vaidehi. So before I stop, I made a mistake of saying first, second, third, and fourth prize for the oral presentation. It's not like that. All these students are awarded for good and best oral presentation. There was no category of first or second or third. Actually, the selection was who was the best in that session, and there were 5 sessions of oral presentation. In every session, who was the best was given award. Okay, so all of them are best presentation. So congratulations, everyone, and thank you. Huh?
Now moving forward with the felicitations, I request Professor Reena Chakrabarti to then on behalf of Professor Reena Chakrabarti, I request Professor A.K. Singh to accept the shawl as a token of appreciation.
क्या हुआ? ये वाला है.
On behalf of Society of Translational Cancer Research, I request Dr. Shijit P. to accept the felicitation. On behalf of Society of Nutraceuticals and Chronic.
One of the society actually authorized us to do this conference. And see for the office they were.
On behalf of Society of Nutraceuticals and Chronic Diseases, I request Dr. Subhash C. Gupta to accept the shawl and memento as a token of appreciation.
दमन सलूजा के लिए मधु चोपड़ा को अनाउंस करना है एंड वो प्रतिमा का वाचन है.
Okay, so no, not the Volvera right now. First, we will try. We will extend our token of appreciation to HCBR, and for Professor Daman Saluja, I request Professor Madhu Chopra to please accept our our token of appreciation, please. Okay, sorry. I now request Dr. Rekha Kumari to receive our token of appreciation for Miranda House for Madam Principal. I now request Professor Alok Bharti to come and do the most coveted job to announce the name of the students for token of appreciation who have actually very actively participated in this conference and without whom this whole conference was absolutely not possible. So before I hand over this to Alok, I on my behalf and from Society of Nutraceutical as well as Translational Cancer Research, from our chief guest and our chairperson and everybody, all the people here actually extend our appreciation for the student volunteers who have been working endlessly, tirelessly to make this event actually a memorable event. Please, the volunteers, stand up so that we can see everybody who participated in bringing this event to this level. Please stand up, everybody. Before I actually short circuited, I'm sorry, probably I'm tired. So actually this is my mistake. And there is another pleasant duty to do. Actually, there are co-conveners of the society, or the conveners of the society who are here, Professor Subhash Gupta and Professor Ajay Kunamakara. They also need to be facilitated. They have been instrumental in, and that is for the society. Yeah. So, okay. So, नहीं. So, लेकिन हाँ तो राजागोपाल को क्यों निकालो? निको रखो ना, दूसरा निकालो. She is totally lost.
Okay, David announced the name. So these are the three co-conveners, or rather I think there are four. There are four co-conveners of this particular conference. The convener was Professor Reena Chakravarti, and they are they are the four main pillars which actually carried out this whole conference, and when we required their help, they were always available. Professor Rajagopal Raman, he was all through the conference, he was there, but because he had to go, so we are just withholding his trophy, and we would like to give this trophy to Subhash. He was running away also. I said that if you run away, your trophy will go. So he has stayed. So he values this trophy more than his engagement. Thank you. You can see that for conference, they are always available. Then Srijit. So Srijit has been, so much instrumental that he was communicating with all the nutraceutical firms in the south. They were trying to get us funds. I think there were two very much important, pillars which actually helped us to get funds and many other clearances. Thank you very much. Okay. Anybody else? And after that, I would also invite Chetkar. Where is Chetkar? Actually, he was engaged in some of the very personal things at home. I had to ask him that please come, because all the show that they could do day before yesterday and yesterday, they were all under the banner of Evolvera. So most of the activities which are associated with students, they are coordinated by one of the very important society which is emerging in our department, Evolvera, and he is still chairing that particular society called Evolvera. So a lot of thanks to them. So before I actually go ahead and read vote of thanks, I would like to continue with a very important token of appreciation for the people who have really contributed. Now it is the individual contributions. The main contribution comes from the session coordinators. So there are 6 very young and dynamic faculties which have joined, or 10 faculties which have joined to our department, and I would like to thank all of them, because earlier we were just thinking that how we can actually engage them, but we were not able to find a consensus. But when we actually started giving them duties, I think all of them came up very well, and they have actually performed all the functions of coordinating these sessions. There were 3 parallel halls that were running and they were really, really very good in conducting these sessions. So all science behind these sessions were actually executed by these dynamic faculties. I'll call one by one. I think Kamlesh is here. Kamlesh is not there, so we'll give Vanika Gupta. Is Vanika there? Vanika is also not there, but Shashank, I'm sure he's here. Please, Shashank, come, and on the behalf of other faculties who are not there, you will be, taking the flags up and high. Okay, now I request Swati Singh if she is there. Swati is there? No. Ananya is there? Ananya was here, I think. Till the end, she was coordinating the session. And then Mamta. Okay, Mamta is a flag bearer. So Mamta is here. Is Ashish there? Can I see Ashish? Okay, Ashish is also not there. Narendra? Is Narendra there? Amod. It's another faculty with us. So he is not there. And Pushplata, oh, Narendra is there. So see how they appear. We think that they're not there. They are always come forward from somewhere, nowhere. Thank you. Narendra was very, very instrumental in one of the earlier our efforts in developing the website. And they and Shashank, I think they helped one of my students very well. I'll take her name. So before we go into the students, I think that we have to also, I request Mr. Nitin Dawar. So whatever show that you are seeing today, all the audio visuals and everything that it's actually conducted by, the successful show is conducted by Nitin Dawar. And he comes from Delhi University Computer Center, and he has seen that there's uninterrupted internet access. We have a very good audiovisual facility here. I really thank to him, and he has been part of the overall purchase committee. So he has been guiding us through the process of purchase with various components associated with the conference. Similarly, Mr. Sandeep Sharma is also there, who had been, oh wow, very nice. I'll come back to him. Okay. Sandeep Sharma also helped us from the general branch. But in the meantime, I said that these people come, that you unko awaaz lagao, or they're always there. So Ashish has also come. So must be involved in some of the good things which are required for the conference. Thank you very much, Ashish. All right. So now it comes please pardon me. I will not take the names of the faculties. Huh? Sandeep Sharma ji aaya nahi hai. Actually, his son is not well, so I had requested him to come. So now I'll take you to the journey with the students. I think they have been, you must have seen, working very hard. And to start with, I think the conference, when it is built up, the most important thing is its website. So it was a big challenge because we wanted to outsource that conference, and Joni was the one which was given the responsibility. And today you can see that single-handedly she has been maintaining this website, of course, with a lot of supervision from Delhi University Computer Center. It's a marvelous and wonderful job, and she could do it very well. Another most important, and you will say that I was always saying that Annapurna, you have heard, but similarly we have Lakshmi and Kuber. So all the conferences require Tan, Man and Dhan. Tan, Man, everybody contributed, but for contributing the Dhan, we had to do a lot of, put a lot of Tan and Man. And this Tan and Man was initially put into force was by Joni itself, and she was fully supported by her junior student colleague, Apurva, and she was marvelous again. So the reason is that she has a control over the conference. She knows from where the funds are coming and where they're going. So we have to ask her permission for spending all those things, whether we have money or not. Okay, now with Apurva, there are a lot of other volunteers: Garima, Gurvinder, Jessica, Mansi, Shivangi, and Juhi. So these were the volunteers who have been working with her as a part of finance as well as welcome committee. Yes, repeatedly. Sab aa jao, fatafat aa jao. Yeah. Yeah, good. So when it comes for a conference, if you are not fed properly, that is not a good conference. If you are actually taken care of, your food is taken care of, and you are living in a very nice, decent environment, very well designed, very well decorated, and all the sitting places are good and well decorated, you will always find it as a successful conference. So the success of our conference relies particularly on tireless efforts of Nikita Agarwal. We had been waiting for her to join. There was a long gap because of some of her personal affairs, but still we were able to get her, and she actually supplemented doubly, whatever time she had taken for her personal engagement, but she could actually compensate all of them in last 1.5 months when she was here. And she was supported by Ashutosh, Anmol, Mukesh, and Gauri Shankar. Till now, you must have been wondering that these names are not new to us, and they're not familiar to us. But the one name which is quite familiar to you will be Tanya Tripathi. Okay, she has been the fulcrum of this conference because she was again single-handedly maintaining the registration of this conference. And we are thankful because it's a very, very serious job, and the calmness and sincerity of her that made it so simple. Nobody has actually found any problem with the registration. She was wholeheartedly, entirely supported by her team with Tanya Sherwal, Neha, Snehil, and Sonal. So they were also very, very competent. Apart from many other her colleagues like Johnny, and almost everyone was helping her. But the main control was again with Tanya. Okay, now it comes to audio visual. So the last 4 or 5 days has been crushing for one of the committee, and the committee is actually headed by none other than Kulbhushan Thakur, but he was also supported morally as well as in all the endeavors by Shashank. They happen to be very close friends, so they were actually running around. So I'm not asking Shashank to come here, but certainly they were both together in all the endeavors. Then they were actually very strongly supported by Anshul. You must have been seeing him. Then Nitish Rawat, Binoy Sahu, Shreyata, Jatin, Anjali, Saima, Kajol, Sana, Rishima. They have been taking all the people from my team, and they are saying that, okay, they are very much required. So they actually manage the show. Whatever, slight problems that occur in the first session, I think they could actually compensate all of them with the help of all this. Please come one by one. Yeah, Saima, please come. Kajol, Sana, Rishima, please come forward. Have one by one. So they are all very, very, they have managed the show very well. Now, another person which I see, I find that she doesn't like to have any other company because that job does not require too many people to be complementing and completing that job. And this girl now, although it's my student, but she very well, is all the teachers of our department actually claims that she is my student. So I this kind of legacy because she is MSc from this department, and most of the teachers they really like her a lot. And one of the teacher which actually claims that she is only my student, so I gave her the charge to interact with her, and that is Namita Agrawal and Divya. I think they both collectively have done a great job of making an abstract book that you see today. And of course, they were helped by Anshul at some occasions and other students also. But if you one day you say that format is like this, the next morning you come, that whole book is now changed into a different format. So I don't know, she works 24 hours, it looks like. So that's why I asked her to report at 4 o'clock in the morning and then leave at 3:30 in the morning. Okay, thank you very much. Now you will see the people who are always out of the hall. I'm sorry, but it's not that they are not interested, but they are more focused to their job. And these jobs are basically looking after the accommodation of all the guests and the transports. The team, I don't know whether I should ascribe Arun for accommodation or Udit for transport or vice versa. So we can just say that accommodation was taken care of by Arun as well as Joni for particularly the the girls' accommodation. Joni was the only one who was allowed to go in, so they were just standing outside. And Udit is always out on the travel counter. Tireless job, and he was very well supported by none other than Rishikesh. Rishikesh has been instrumental in setting up our transportation. Where is Rishikesh? Without Rishikesh, Udit is incomplete. So there are other two Abhisheks are there. You will not be able to guess who is who. So there are two Abhisheks. And then there is Deblina. और एक तो जानबूझ के अपना नाम भी लिखवाई है, जोनी यादव जरूर भूलना मत. ठीक है. So Deblina is also very enthusiastic in this. Now I will request the students who are involved in cultural event. Yesterday the food was quite tasty, so all the guests, without actually thinking that we have to honor the students who are involved in this cultural program, they just ran away. So we thought that we will postpone it and withhold again all the guests. So here in this case, I will request all the cultural event students, please come to the stage and we will be giving you a small token. अनुराग, प्रतिभा, मैंने इनको बोला था लाइन लगा के रखो, बहुत देर लग जाती है, आप ढूंढते रहो. ठीक है. So I'll take Anurag, Pratibha, Aman, Sheetal, Sonal, Gauri, Mansi, Jessica, Rishima, Monica, Parul, Chetkar, Poonam, Deblina, Manvi, Kushagra, Chandrakant. So we have a small token of appreciation for them also. (14) Rangoli and other volunteers. I think that everybody wants to have a photograph in front of Rangoli. So this particular job was basically executed by a group of people: Astha, Payal, Niharika, Seema, Pratibha, Shivangi, Survi, Vikas, Minakshi, Devanshu, Gordon, Divya, Tejveer Singh, and Senrang. And lastly, but not the least, they were actually supported. All this activity was supported by our lab attendant, Anshul, Ravi, and Prem. If they are they're in the hall, please, they can also come forward. Please excuse me if I have left anyone. It is not intentional. So you can still be appreciated. You just let me know. I'll see how can I compensate this loss. Okay. So with this, we stop the token of appreciation, and I will request Professor. Oh yes, I think we had. There was one other delegate, Mr. Mahesh. extempore to actually deliver the Saraswati Vandana in the beginning of the conference. So he deserves a great deal of great applause. Now, at the end, I request Professor Bharat Agrawal. We have a small memento for our chief guest to give that memento. Can you just please bring that? So we have got a memento for you, sir. Hopefully, it will be there on your side table always, and keep blessing you. Nikita? Is Nikita there? All right. So in the meantime, when this memento comes, let me read a vote of thanks. That was the main thing. In fact, whatever I had done till now, giving the token of appreciation, it's a big vote of thanks. But it is a formal one. I would like to, so first of all, thank all the speakers and chairpersons for making out their valuable time for this particular conference. We are also thankful to the head of the department for for keeping us always keeping her always available for this particular important event. We have already seen there are 3 different societies which are involved in this particular event. There were Society for Nutraceutical and Chronic Diseases, Society for Translational Cancer Research, and then there was Evolver was involved, and we are all thankful to them. We are also thankful to the students who have actually participated both online as well as offline. And this is a very, very enthusiastic participation post-COVID. And then the most important thing is the money. I am really thankful, whatever effort that we made, we had a significant amount of, funds so that we can have a very successful conference. And apart from that, I think the people you might not have been seeing here is the non-teaching staff. So many of the non-teaching staff have been the instrumental in taking care of this activity on day-to-day basis. And you have already seen that all members of Molecular Oncology Laboratory and Tumor Immunology Laboratory, and there is also laboratory from of Professor Namita, they have been tirelessly involved in this activity. And we are very much thankful to all of them. In fact, you might have seen that in whole program, all the teachers and faculties of this Department of Zoology were involved, and our sincere thank to all the faculty. And we have the organizing committee was not only having Delhi University's Department of Zoology people. There were a lot of people coming out from not only University of Delhi, they were also coming from other places. So like ACBR, and as well as many places in India, many institutes of India. We all are thankful to the main organizing committee. And contribution and solid support of Engineering and Computer Center is also commendable. And last but not least, I thank everyone for cooperation in making the function a resounding success. And hope you will keep all good memories despite there might have been some inadvertent hiccups, errors and omissions. We look forward to meeting you again. Okay, and with this, I draw your kind attention. We are going to felicitate our chief guest. Please go ahead. Yeah. Once again, I thank you all for your attention, and
Let's call the day with high tea. Before we disperse, please let us stand for the national anthem. Please, आप भी आ जाइए.
जन गण मन अधिनायक जय हे भारत भाग्य विधाता पंजाब सिंध गुजरात मराठा द्राविण उत्कल बंग विंध हिमाचल यमुना गंगा उच्छल जलति तरंग तब शुभ नामे जागे तब शुभ आशीष मांगे गाए तब जय गाथा जन गण मंगलदायक जय हे भारत भाग्य विधाता जय हे जय हे जय हे जय 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 हे.